It is always a little exciting and also scary when we can fit the entire rest of our schedule onto the whiteboard, and yet here we are. We have just a handful of classes left, and we are done with the semester. Doesn't mean we don't have a lot to do over that time, as you can see. Today, we're going to continue our discussion of neurophysiology and try to catch up some on our nervous anatomy that we fell behind on in the last class. I know I threatened to start with the anatomy first, but I think we're still going to do the lecture part and then get that out of the way and then get as far as we can in the anatomy. Yeah, the handful of assignments left. Again, I appreciate it is the day before Thanksgiving, and I'm sure like you have some prep to do. We're not even hosting this year, and I still have a fair amount of prep to do, but at least we have a morning class and we're done by 9:30, and so that should give us plenty of time. So despite the fact that I know many of you have kids who are off this week, and again, other plans and travel and all those things you want to do, we are going to have a 5-point daily quiz, which hopefully won't make or break your grade in this class, but I did want to make you aware of this one because it is on a day where I know a lot of people tend to not want to be here. But more importantly than that, we will be reviewing the cranial nerves in class, as I mentioned, and that could easily be 25% of our lab exam. And anything that important will probably bleed over into the lecture exam as well. So this is a huge part of the nervous system, and I want to make sure you guys are forewarned and encourage you to be here to ask questions and make sure you understand this material. One more Physio X. This one's the biggest with 9 activities. So again, don't save that for Sunday night to start working on that because you won't get any sleep because, as you know, it is not hard, but it is time consuming. So do that. One more assignment that is graded for correctness, and that is your nervous review, which is due Wednesday. I thought we had something else on Wednesday as well. We do. But 15-point nervous system review, which is going to be graded for correctness. So again, that is really about emphasizing the relationship between structure, function, and location. Now, a lot of that involves the spinal cord, which we have not talked about yet, but we should hopefully either get to that today or Wednesday at the latest. So that should give you plenty of time, a full week, even if we don't get till Wednesday, to make sure you've mastered and understood that information. Monday the 6th, we have 2 assignments that are due, a unit review, which you know how to do. You can kick that out without any problems at all. The last activity, the last homework assignment is hopefully a fun activity. It is a Reflex Lab, something that we do in the classroom. I appreciate that you are not going to have all of the resources at home to be able to do this. Most of you have a flashlight or a pen light or something you can do to flash in each other's eyes, but you probably don't have a tuning fork or some of those other types of resources like that. But even without those things, you should be able to do most of the activities, and those that you can't, I want you to do as a thought experiment. Don't just say, I couldn't do this activity. I want you to at least write down what you think would occur. And there are basically 2 procedures. Procedure 2 on pages 371 through 374, and then also procedure 2 on page 380. So even if you can't do all the activities, I want you to at least pretend you could do all the activities and tell me what you think would happen. Don't just say, I didn't have a tuning fork, we didn't do this. Figure it out. And then it's exams. Wednesday the 8th, we have our last lab and lecture exam, again, a Wednesday exam. So that Monday, we will be getting some new information right before the exam. I'll try to limit how much of that there is. That also means that the 4th and 5th is your big weekend to study that. And that should be your focus, because then after that, you have a full week until your cumulative final exam. We do not meet on the 13th. That is finals week. So some people may have finals on that day. So we don't have class scheduled. So you've got a full week off to study and look back at the material that we've covered in this class to prepare for that cumulative final exam. It is 100 points. It is 100 multiple choice questions. Again, the huge advantage of that is there's no essay questions. So the correct answer is right there in front of you. You just have to recognize it. So there's a big difference between pulling the information out of the ether and recognizing it on the screen in front of you. So hopefully that will help you to be successful on that. And remember, it counts as a grade on its own, but it also has the opportunity to replace your lowest lecture score. So for some of you who stumbled on one of the exams, this could be an opportunity to really help to boost your grade to make sure that you get the grade that you want. And then on the 16th, you get to sleep. All right. Questions on any of that? That's all that's left. Oh, Ava, I don't know if you're talking, but you look like you're talking, but I can't hear you.
Weird.
There you go. Now I can hear you.
Now or no?
Yes.
Okay. I was wondering if on our last Zoom day, if we have extra time, could we do a little bit of a review for the final?
Yeah, always.
Oh, okay.
And even though we don't have class on Monday, the 13th, I may set up the time to have a review for the final exam, so we can probably set something like that up as well.
Okay, thank you. That'd be great.
Yes. And Mitch, yes, the final exam will include everything we've covered in this class. Remember, lab and lecture really aren't two classes, it's just different modalities for studying the material. And when we're online, that kind of goes away a little bit. It'll be similar types of questions to the ones you've seen before. You may see one or two of the previous questions. I don't pull the pool just from the previous ones, but I don't preclude using them either. But yes, things that there may be some pictures of, the histology of a bone, for instance, and you have to recognize a concentric lamillae or something along those lines. So there could be a small amount of lab stuff as well, but it'll all be, like I said, it's all multiple choice questions. All right. Any other questions?
Is the final curved?
Typically not with 100 multiple choice questions. Usually it doesn't need to be curved. If there's something odd happens and the grade seems particularly low for some reason, especially because of the randomness of it being online, I'm not opposed to curving the final, but often it doesn't need to be curved. So usually the grades on that are as high, if not higher, than you see on the normal lecture exams. So unless something wonky happens, there isn't a need to curve it, no.
Just out of curiosity, is it like 1 point each, so it's 100 questions, or is it like 50 questions?
Yes, 100 multiple choice questions, and you'll have 2 hours to complete it. All righty. All right, any other questions? All right, let's pick up where we left off then. We have been working our way, oops, wrong button, through the physiology of a neuron. We now know out here on the cell body and on the dendrites how to produce a graded potential. At our axon hillock, we know how to produce an action potential on both a myelinated and on an unmyelinated axon. So I'll myelinate it on one side and leave it unmyelinated on the other. We've learned how to propagate an action potential. So really the only thing we haven't talked about what a neuron does is actually the very first thing we learned about a neuron. And that is how that neuron communicates with its target cell. And what do we call that specialized area of communication between a neuron and its target cell?
Synapse.
Exactly. A synapse. So the last thing we have to talk about to understand how a neuron functions is the part that we learned first and the part that we probably know the best. How a synapse works. A synapse, remember, is the functional junction between the presynaptic neuron and whatever its target cell is. Now, just out of curiosity, what do you think the most common target cell is? What do you think most neurons talk to?
Other neurons?
Yeah, exactly. The most common target cell is another neuron. Most neurons talk to other neurons. If you remember, if you think about it, over 90% of our neurons are in the central nervous system are interneurons. And interneurons are basically the ones that receive information from sensory, process that information, and then send that information to motor. So not surprisingly, the most common target is another neuron. So most neurons actually talk to another neuron. Because of that, we use terms like presynaptic neuron for the one that's doing the communicating. And so what do you think we would call that target cell that receives the information then?
Target cell.
Postsynaptic.
We'd call it a target cell, but we'd also call it a postsynaptic neuron. All right, so there you go. Simple terms. Now, we have not used the term this way. However, there are actually two types of synapses, and we have talked about both of them. The first type of synapse is actually an electrical synapse. We talked way back in chapter 3 about gap junctions. If you remember, gap junctions are made up of special proteins. Those special proteins were called connexons, right? And they were transmembrane proteins from one cell that would bind to the transmembrane proteins of another cell. And they basically would form a fluid-filled pore that connected the two cells together. All right. So this gap junction is really what we can also refer to as an electrical synapse. Basically, what we've done, just like your electrician in your house, we have hardwired the two cells together. As we know, an action potential is primarily caused by a huge influx of sodium ions. And if one of these cells receives a huge influx of sodium ions, what do we know those sodium ions are going to do? Do they just stay in the cell?
They're gonna diffuse into the other.
Yeah, they're going to spread. And as they spread to the next one, this cell would then reach threshold. And when it reaches threshold, it would fire an action potential, because as we mentioned, the most common target cell is another neuron. So it could reach threshold and fire an action potential. It's like I said, it's basically hardwiring these cells together. What's the advantage of something like this?
The cells are able to work together as one.
Yeah, the cells work together as one. What else?
It allows communication both ways.
Absolutely. Mitch has got it first. It's fast, very fast, right? Just a simple spread of sodium from one side to another. Absolutely. And Ava, you are also correct. Just as easily as the cell here on the left could have the huge influx of sodium and it spread to the right, it could work the other way as well, where it has the huge influx of sodium on the right and that spreads to the left. So the advantage of this is it allows for two-way communication. All of these sound like really, really huge advantages. And yet, most neurons do not use this type of synapse. Why not? If it's fast, if it allows them to work together as one, if it allows two-way communication, those all sound like really important, really huge, really useful things. So why wouldn't our nervous system want to use it? There must be some huge disadvantage.
You can't modify it.
Right, I like that. Absolutely. You and who is it? Mitch both absolutely hit it on the head. right? There are times when we don't want them to fire together. The disadvantage of this is that they always work together. And there are times where we don't want this. We want the system to be modifiable, right? You don't always want to say, you don't always want to respond the same way to the same stimulus. Me on camera, holding a red pen, looking through the camera at you in the eye and saying, I'm going to kill you. And a bear with an axe walking into your room, looking in you in the eye and saying to you, I'm going to kill you. You are receiving the exact same stimulus from both of us. Do you want to respond the same way to both of us? No, absolutely not, right? We want to be able to modify this system. We want to be able to make decisions, to change our behavior based on all sorts of different types of things, right? Even our quote unquote reflexes, right? If I have you sitting on a table in my office and I hit you in the knee with a hammer, what's going to happen?
Our knees.
Yeah, Aubrey's got it. Your leg's going to kick up. Absolutely. However, if I then put a sleeping baby in front of your foot and I hit you in the knee with a hammer, are you going to kick the sleeping baby? You horrible, horrible person? No, of course not. Even a reflex, we can modify the behavior of, right? So that ability to be able to change, in our nervous system, we want to be able to make decisions. We want to be able to modify behavior. And so while there are lots of places where these gap junctions, these electrical synapses are really important, muscle, our cardiac muscle, we want our heart to work together as one. Our smooth muscle, we want the parts of our stomach to work together as one. And there are a couple places in the nervous system, but most nervous system synapses are not electrical, because we want to be able to make decisions. We want to be able to modify behavior. So again, this isn't new information. We know what a gap junction is and how it works. We've just given a fancy name to it now, that it's an electrical synapse. All right, so we talked about those. A chemical synapse is the synapse that we've kind of already talked about in this class, that when we first said the word synapse, right? What are the three components of a chemical synapse?
The synaptic end bulb.
Excellent. We have
Of
a
the
synaptic end bulb,
presynaptic cell.
the presynaptic cell, absolutely.
Synaptic cleft.
A space in between. So we have our synaptic end bulb of our presynaptic cell. We have a space in between, they don't touch. And then obviously we're going to have our postsynaptic cell. And on this postsynaptic cell, we don't know what the specializations are going to fully be, but we know we're going to have some type of chemical receptors, right? That will receive the information from the neurotransmitters that are released from the synaptic end bulb. So since we know most neurons talk to other neurons, that means this is going to be a dendrite or a cell body. And that dendrite and that cell body is going to have some kind of chemically gated ion channels that will allow it to be able to be communicated. I don't know how often I have shown this. Probably seen it as we bounce back and forth between things. I don't even know if I can show you now that I think about it. Yeah, I don't know if I can. Hold on, let me see if I can find it this way. It's easier in the classroom. I know you guys get to see it there. There it is. My desktop in the classroom and in the on my computer here is this image here. The scientific journal Science every year does a contest where they do the best illustration of a physiological process. And many years ago now, gosh, probably close to 20 years ago, this was their $10,000 winner. And as you can see, this is a neuron in the middle of communication. You can see that we have our synaptic end bulb here with its yellow action potential coming down. Here's our mitochondria and our synaptic end bulb and our vesicles. And it is getting ready to release a signal into that synaptic cleft to another neuron. And this structure, this enlargement of the dendrite is called the dendritic spine. So we have this dendritic spine that is getting ready to receive that information. So we have that synapse where that information is getting ready to be processed. So again, this is a beautiful illustration. Being a neuroanatomist, it's pretty much my background of everything. I don't have pictures of my kids on my computers. I have pictures of this on my computers because, well, quite frankly, I like neuroscience more. But the point being that, oops, well, I've been lost it all because we were here. As we know, that chemical synapse basically has three components to it, right? The cells don't touch. So we have some type of presynaptic, well, we have the synaptic end bulb. The cells don't touch. So we have a synaptic cleft. And then we have that target cell with some type of chemical receptors on its surface. All right. It's called a chemical synapse because, as we know, our presynaptic neuron, let's go ahead and name these things. A presynaptic neuron is going to release some type of chemical signal, a neurotransmitter, which will bind to receptors on the opposite side. And this takes time. As we know, we need to let calcium into the synaptic end bulb. That calcium then builds up and triggers the exocytosis. Our neurotransmitter then has to diffuse across the synaptic cleft before it can bind open channels, allowing something like sodium to come inside and get enough sodium inside where we reach threshold. This is a much longer process than just having sodium move through an electrical synapse, right through that gap junction. Now, again, I say relatively slow. We're still talking about half a millisecond, but it isn't the instantaneous flow of ions that we see in that electrical synapse. So it does take a little bit of time. Also, unlike the gap junction, this only allows one-way communication. Our target cell can't talk to the synaptic end bulb the same way that we saw in the the electrical synapse, where it can go in both directions. But the advantage of this is that it is modifiable. As we've talked about, depending on what type of channel you open, you can make the cell either more positive on the inside, which we would call excitatory, or we could make it more negative on the inside, which would be inhibitory. So we can get different effects. We can modify the behavior. And this is especially true in neurons. Neurons, a typical neuron receives 10,000 different synapses, inputs from 10,000 different neurons. So it is receiving a massive amount of information. Basically, neurons are accountants. They're bean counters. They count how many positive things they come in, they count how many negative things they come in, and then they determine what they're going to do based on those two pieces of information. So they're able to receive lots of signals. And because of that, we are able to modify our behavior, right? It doesn't have to produce an action potential. It may produce an action potential. We get to make decisions. We can change our behavior. 3 different people can walk out of stores carrying their bags of groceries. And for all 3 of those people, the bottom breaks on the bag, all their groceries pour out, and those 3 different people could all respond to it differently. One person may fall to the ground crying, another person may sigh just walking away, and the third person may just, shrug, grab a cart, throw all their stuff in there and go to their car. Heck, the way you responded to that could vary depending on what the day is, right? If you just had a great breakfast, your boss just gave you a raise, right? You could laugh hysterically at that happened. Whereas if someone just finished breaking up with you, right, you could weep endlessly just because your bananas fell to the ground, right? So again, there are lots of decisions, lots of modifications that are possible based on this. And the best part of this is there's really no new information. Once again, on this exam, I could ask you how a chemical synapse works, but you already know the answer to that. It starts with a neural action potential spreading down the axon to the synaptic end bulb, depolarizing the synaptic end bulb, opening voltage-gated calcium channels, allowing calcium into the synaptic end bulb, which triggers exocytosis of our neurotransmitter into the synaptic cleft. That neurotransmitter diffuses across to the target cell, where it binds to a chemically gated channel, opening that channel and allowing ions to either move into or out of the cell. And of course, that positive ion, or that ion, pardon me, that charged ion, moving into or out of the target cell, is going to produce some type of graded potential. All right. As we know, graded potentials can be positive or negative. They can vary in size. Right? The more channels you open, the more ions are going to flow, the bigger that's going to be. And since we are doing this in the target cell, and that target cell is usually a neuron, we can give a fancy name to this graded potential. We can call it a postsynaptic, because this is the postsynaptic cell. So a graded potential in a neuron, we can call a postsynaptic potential. And as we hinted at, there are 2 types: positive and negative, or excitatory and inhibitory. So we can have an excitatory postsynaptic potential or an inhibitory postsynaptic potential. What do you think the obvious difference between an excitatory and inhibitory postsynaptic potential is?
One's positive and one's negative.
Yeah, which one's going to be positive, do you think?
The excitatory.
Yeah. There we go. I appreciate I went through this chemical synapse really fast, but you needed to know it for the last exam, so I didn't think we needed to spend too much time on it. But I'm happy to do that if you need it. Does anybody need a refresher? Need me to go a little bit more slowly between what's happening here at the synapse?
No, but I have a question.
Yeah, absolutely.
So for this one, so the only real difference I'm seeing is that instead of the acetylcholine, it's causing the neurotransmitters to diffuse.
Okay. Yep. Well, so you're right. There are two differences. Remember, neurotransmitter is the general term for the chemical signal that is released from the presynaptic cell. Acetylcholine is a specific neurotransmitter. In fact, it is the most common neurotransmitter in the peripheral nervous system.
Okay.
However, when we're talking about this neuron to neuron communication, we're most likely in the central nervous system. And so it probably isn't going to be acetylcholine. And since it's not acetylcholine, that's why we're using the generic term. Okay.
Yep.
So absolutely. So that's where that's the difference. And acetylcholine is a type of neurotransmitter, but it's not the one we're likely to use for neuron to neuron communication. So that's why we use the generic term. In about a half an hour, we'll identify what neurotransmitters they might be. But again, for right now, we can just use the generic term neurotransmitter. The other big difference is in the target cell. Obviously, when we talked about the neuromuscular junction, the target cell was the skeletal muscle cell. And specifically, we were communicating to the motor end plate. Right? Here, we are talking to another neuron. So we're going to talk to the dendrite or the cell body. Notice the picture, it's at the cell body right in between the dendrites that it is talking to. And there's going to be some type of chemically gated channel. Remember, in the skeletal muscle, we knew exactly what that chemically gated channel was. It was a chemically gated cation channel that allowed a lot of sodium in and a little potassium out. In our neuron, there are lots of different types of chemically gated channels. So we don't necessarily, when we're talking about it in general, we don't know which one it is. So we just, again, we're using the generic term neurotransmitter, we're using the generic term chemically gated channel. Because they can be different. They're going to be different from cell to cell to cell.
If we explain it this way on the 4 steps that they have here on the exam, will we get full credit, or do you want us to go into more detail?
Again, I don't care about the number of steps you use, but remember, it's always about detail. So I want the same level of detail that we used when we were discussing it for the neuromuscular junction. You've already learned the neuromuscular junction was just one specific example of a chemical synapse. So that is the perfect example of how you would describe how a chemical synapse works. So I'm looking for the same level of detail that we talked about it for the last section, for this section.
Like the whole process from like start to finish, like from initiation to, that's where I guess it confused.
Well, again, we don't, I see what you're saying.
Could you just possibly go over it then? I don't mean to be annoying, but.
No, you're not annoying at all.
Okay.
So again, I understand the question you're saying. Obviously, we're not going to talk about excitation, contraction, coupling. Obviously, we're not talking about the contractile cycle, so that's true. But you're also correct. We're not going to talk about terminating the process or modifying the process. We're just talking about initiation of this process. So if on the exam, I ask you, you get the essay question, describe signal transmission at a chemical synapse. It would start with a neural action potential spreading down the axon to the synaptic end bulb. Right. From there, it is going to open the voltage gated chemical, depolarizes the cell. I guess we have to start with that. It depolarizes the synaptic end bulb. It opens the voltage gated calcium channels. Once the voltage gated calcium channels open, calcium enters the synaptic end bulb. The calcium inside the synaptic end bulb triggers exocytosis of the neurotransmitter. Now, obviously, I'm abbreviating all these things. You won't be abbreviating them on the exam, but you get the idea. Triggers the exocytosis of the neurotransmitter into the synaptic cleft. Once in the synaptic cleft, the neurotransmitter diffuses to the target cell. Once it diffuses to the target cell, it binds to chemically gated ion channels. Ions are going to flow into or out of the target cell, and that will produce a postsynaptic potential, a graded potential in the target cell. And as we know, if this is a neuron, then if this target cell was a neuron, if that graded potential is positive enough to spread to the axon hillock, then it has the potential to produce an action potential. I don't think that necessarily has to be part of our explanation of a chemical synapse, but if we think back to the very beginning of this, that's exactly what's going to hopefully happen. This graded potential will produce an action potential, and this neuron then sends a signal to the next neuron. And the process continues.
Thank you.
Yep.
I have a question.
Yes.
Would I get marked down if I put a chemically gated cation channel? Because you put ion channel.
Well, because remember, the cation channel was specific to the neuromuscular junction. This channel here could be a chloride channel. If it was a chloride channel, then we would be inhibiting this cell. So honestly, for the exam, no, but I prefer you be more generic because it's going to lead into what we're going to talk about next. The key to what we're talking about next is the goal of this communication, as we've talked about, is to produce a postsynaptic potential. But the way we modify this is, as we've talked about, some will be inhibitory and some will be excitatory. We don't always want it to be excitatory because then we might as well just have them hardwired together. So honestly, if you slipped and called it a cation channel, I wouldn't ding you on the exam.
All right, thank you.
But it is important to understand that it's not always going to be a cation channel. There's going to be different types of channels that it communicate with, which is going to produce different types of postsynaptic potentials, which I believe is where we're going next. So again, the binding of the neurotransmitter to the postsynaptic potential is going to open some type of ion channel where different ions can flow in and out. And again, let's just think of the 4 examples we've talked about? What are the 4 main ions we've been talking about in this class?
Sodium,
Sodium. What else?
calcium,
Calcium. What else?
potassium,
Potassium. What else?
chloride.
All right, there you go. If I opened a sodium channel, which way would sodium flow?
Into.
Yeah. And what effect would it have on the cell? Would it make the cell more positive or more negative?
Positive.
Excellent. What happens when calcium goes into a cell? Well, calcium goes into the cell, but what happens typically when calcium goes into a cell?
It makes it more positive. It depolarizes it.
True, it does depolarize it. But what else do we know about calcium?
Those wonky things.
Yeah, it makes cells do wonky things. And again, making cells do wonky things is something very special. So typically, if we're just trying to make the cell more positive or more negative, we're not going to want to use calcium because of calcium's potential to do wonky things. So that leaves us 3. Which way does potassium move?
Out of the cell?
Yep. And when it moves out of the cell, what does it do to the cell?
Makes the cell more
Yeah, it makes the cell more negative. And then which way would chloride move?
into the cell?
Excellent. And what effect would that have on the cell? Whoops.
More negative?
There you go. Excellent. So depending on which ion moves in or out, we can actually make the cell more positive or we can make the cell more negative. And again, these ions flowing in and out produce a graded potential. Notice it can be positive or negative. And the number of channels we open determines how much of the ions flow. If we just open one channel, we're just going to get a little bit of ions flowing and we're going to get a small signal. If we open a lot of channels, a lot of ions are going to flow and we're going to get a bigger channel, a bigger change. So again, we can get that graded potential, which varies in amplitude and can be positive and negative. So that is going to be important. And like I said, not any new information, just a new name. Because this graded potential is occurring in a neuron, we call it a postsynaptic potential. So if the neuron is a target cell, let's actually say it that way. If the target cell is a neuron, we call its graded potential a postsynaptic potential. And the reason for that isn't just because anatomists hate you and they want to give a separate name to things, but when we talk about a postsynaptic potential, we can talk about them being excitatory or inhibitory. Yes, excitatory means more positive. Yes, inhibitory means more negative. And again, notice we have those other terms we've talked about, depolarizing and hyperpolarizing, moving it up from resting membrane potential, moving it down from resting membrane potential. But if you think about it, what we care about is if the target cell, the neuron, fires an action potential. That's really when our target cell is a neuron, what we care about is it going to fire an action potential? And so an excitatory postsynaptic potential means that the target neuron is more likely to fire an action potential, which is why we call it excitatory, whereas inhibitory means that the target neuron is less likely to fire an action potential. I know this can get a little confusing because we're adding all this vocabulary, but these are not new concepts. Let's get away from the words because words are confusing, and let's look at pretty pictures. Here is an example of an excitatory postsynaptic potential. Here we are measuring the membrane potential of the target cell. Notice that target, let's be even more specific, neuron. That target neuron was happily sitting at -70 millivolts, and then we gave it a stimulus, and suddenly it got closer to threshold. Now notice your book, or not, this isn't actually from your book, this is from a different book, gave us a different threshold. So let's cheat and change that to 60, because that's what we're using. There we go. Notice this postsynaptic potential got us closer to threshold. Didn't reach threshold, but it got it closer to it. And so this would be an example of an excitatory postsynaptic potential. It causes a depolarization, makes the cell more likely to reach threshold. Notice that key phrase, more likely. Does it have to reach threshold? No, it doesn't have to reach there. It does not have to get there, but it just has to get it closer. If it makes it more likely to reach threshold, then it is excitatory. Now, based on what we talked about in the last page, what type of ion channels are we most likely going to use for excitatory postsynaptic potentials? What kind of ion channels? Say again?
Sodium.
Yeah. Remember, opening sodium channels were the ones that made the cell more positive. So these typically involve sodium channels. Conversely, an inhibitory postsynaptic potential is one that causes the cell to hyperpolarize, get more negative than the resting membrane potential. Notice when that happens, it makes it less likely, it makes it harder to reach threshold. Not impossible necessarily, but it makes it harder to reach threshold. So it doesn't have to 100% stop the action potential. It just has to make the action potential harder. And if it makes it harder to reach the action potential, we call it inhibitory. And which ion channels are we most likely going to want to use for inhibitory postsynaptic potentials?
Chloride and potassium.
Exactly. So this typically involves chloride or potassium channels. Questions on this? I have a question. Look at this change in the membrane potential. It's bringing the cell way down more negative, nowhere near threshold. So why do I say less likely? Why don't I just say that it doesn't reach threshold?
Because neither have to reach threshold.
True, absolutely. But let's go back to one of the points that I made briefly, but maybe didn't emphasize. How many synapses does a typical neuron have on it?
More than one.
Yeah, in fact, if you remember, I said 10,000. So if one neuron is releasing a neurotransmitter that opens up a chloride channel and makes the cell more negative, but the other 9,999 are opening up sodium channels, do you think that cell's likely to fire an action potential?
Yeah.
Yeah, right, absolutely. So notice this one made it more negative. It is inhibitory, but it didn't stop it from firing an action potential, right? As I mentioned, neurons really are accountants. They're bean counters. Think of it this way. If a neuron received 5,000 excitatory postsynaptic potentials and 5,000 inhibitory postsynaptic potentials, what do you think is going to happen to that neuron?
Nothing.
Nothing. It is receiving a massive amount of information, a massive amount of ions are moving, and nothing happens. Because basically, one excitatory postsynaptic potential cancels out one inhibitory postsynaptic potential. They really are accountants. You have $10,000 coming in and you have $10,000 coming out. You're not any richer than you were before, right? Your goal is to reach threshold. Your goal is to buy that plasma TV in time for Thanksgiving because there's going to be all those football games on. Right? You need, your threshold is $1,000. You need $1,000 to buy that TV. And yes, you have $10,000 coming in, but if you spend $10,000 on food for Thanksgiving, you haven't made any advantage. You haven't gained anything, and there's not going to be any change. And that's really what neurons do. They get all these positive, all these negative inputs to them, and that's how they make a decision. They count the beans. Do I have more positive than negative? Is the more positive than negative enough to reach threshold? Then boom, if it is, I fire an action potential and the signal moves along its way. And that's really the key to this. One excitatory postsynaptic potential is not enough to generate an action potential. Remember, excitatory postsynaptic potential is just a fancy term for the graded potential. And remember, we said the average graded potential was only about a positive or negative 2 millivolt change. Is 2 millivolts going to be enough to reach threshold?
No.
No. So typically one can't do it by itself anyway. But like we said, a typical neuron can have as many as 10,000 inputs to it. And so it counts beans. Is there more excitatory? Is there more inhibitory? And the goal is to reach threshold. Now, let's make this even simpler. Let's say, for simplicity's sake, that one excitatory postsynaptic potential equaled 1 millivolt change. If I wanted to reach threshold, how many excitatory postsynaptic potentials would I need?
60.
Well, but remember, I'm starting at -70 and I need to get to -60. So how many would I need?
10.
10.
Sorry.
Right? Your book uses a great analogy, which I absolutely love. You have a bathtub that it takes 10 gallons of water to fill. So we need our 10 excitatory postsynaptic potentials into that bathtub to fill it up, to reach threshold, to fire an action potential. But here's the problem with the bathtub. The problem with the bathtub is that I've lost the drain cover. So my bathtub is, of course, up here on the second floor of my house. And of course, the faucet is in the basement. So if I have to start in the basement, filling my hot water in the basement, come up here 2 floors, 3 floors, up to my second floor, dump the bucket into the bathtub, go all the way back down to the basement, fill that bucket again. By the time I get back up to the second floor, what's happened to the first gallon of water?
It's drained.
It's drained.
Yeah, it's drained away. I'm not going to be able to fill that fill that bathtub by taking that much time. So notice, not only do I need these 10 excitatory 10 excitatory postsynaptic potentials to reach threshold, but they have to be close in time. We need to be able to add them together quickly. And there are 2 ways that we can do this. One is I could have the faucet right next to the bathtub. If I had the faucet right next to the bathtub, and maybe I had 2 or 3 buckets, I could be dumping one bucket while a second bucket is filling, and I could grab that and I can dump it. And maybe me by myself, if I was able to do it fast enough. Or if I got all 10 buckets, filled them in the basement, brought them up and sat the 10 buckets next to the bathtub. And then me by myself, I could dump all 10 of those into there. Could I maybe get them in fast enough to be able to fill that bathtub up and reach threshold?
Yeah.
Possibly.
Yeah, probably, absolutely. So if I did it fast enough by myself, I could reach threshold. But I'm sure, like you, whenever I plan on taking a nice hot bath, I like to invite 9 of my closest friends over as well. And so the 10 of us could all be standing around that bathtub with each one of us holding a bucket. And if me and 9 of my closest friends all dump our buckets at the same time, can we fill the bathtub up that way?
Yes.
Absolutely. So notice there's not just one, but there are 2 ways to fill the bathtub. And those 2 ways are spatial summation and temporal summation. Temporal summation was the first example I gave you. Me doing it all by myself. We have one synapse that fires rapidly. And as it's firing rapidly, we are able to basically do our wave summation. This is basically similar to wave summation, right? One synapse firing rapidly adds its action potentials together, or more specifically, its excitatory postsynaptic potentials together. And if it does it fast enough, we can reach threshold. So that is me dumping all the buckets by myself. However, if me and 9 of my closest friends all are in the same location, Notice here, the key is temporal. I have to do it all close together in time. But if me and 9 of my closest friends are all together in the same space, in the same location, and we all fire action potentials at the same time, we can here, the goal is to get multiple action potentials, to get multiple excitatory postsynaptic potentials. But here, it is not me doing multiple signals myself. Here, it is multiple neurons that all fire in the same location. And when they all fire in the same location, we're able to add excitatory postsynaptic potentials together to reach threshold. So the goal is the same. The goal of both is the same. Fill the bathtub. Get a big enough signal to reach threshold. And either one neuron can do it by itself by firing very rapidly, or we can have 10 neurons that all fire one action potential at the same time in the same location and get it to reach threshold that way. And that determines whether or not our neuron fires an action potential. Great question. Let's go back to the example of the reflex. Again, reflexes are so simple, they're sometimes really great and easy to use. When you hit the knee, and we'll actually talk about spinal reflexes and why this happens, but basically the whole point of hitting your knee and having your leg kick out is the doctor's way of checking to make sure that your muscle-related neurons are working properly. That when you get a specific stimulus, like basically that's when he hits you in the knee, it's tricking your leg into thinking that you're falling over. And so you kick your leg out to get your balance. So when he hits you in the knee with the hammer, all of the neurons associated with that stimulus are going to the neuron that controls your muscle, saying, fire, fire, we're falling over, fire, fire, fire. But then he puts that sleeping baby in front of your foot. And the neurons in your eye, seeing that baby, go, holy crap, I'm about to kick a baby. And they send negative signals to that same muscle, saying, I know you want to contract, I know you think we're falling over, but if you contract, you're going to kick a baby. And if that happens, we're going to end up on social media and we're going to get canceled and it's going to be horrible. So don't kick the baby. And so notice then that neural input from your nervous system, from your brain, from your eye are saying, no, 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 don't do it, don't do it, don't do it. And you override the reflex and you don't kick out. So that muscle is getting two conflicting signals. One saying, yes, yes, we're falling over, we're going to die, fire. The other saying, no, no, you're going to kill a baby, stop, and inhibitory. And so two conflicting signals coming in. Hopefully the neural one, the visual one is stronger so that you don't kick the baby. That make sense? Excellent. All right, how much more do we have left of this? All right, I know we'd normally take a break, but we only have 2 more slides. So let's finish this up and then we're actually done with the lecture for today. Okay, so notice as we've talked about, the key here is communication. We have our neuron that is communicating to another neuron by releasing a neurotransmitter. Now, over 100 different neurotransmitters have been identified. So far, we've learned about 1. acetylcholine. We'll talk about a handful more in just a minute. Some you're familiar with and some you may not be. And the real key is when we talk about a neurotransmitter, remember we talked about how acetylcholine, oh, and let's actually enhance this drawing. When the target cell was that skeletal muscle, and we released acetylcholine, what effect did that have on the skeletal muscle cell?
Excitatory?
Yeah, excitatory. Remember,
Muscle
one
control.
neural action potential basically produced one muscle action potential. So it was excitatory. It made it contract. So would that make acetylcholine excitatory or inhibitory as a neurotransmitter?
Excitatory.
It'd make it excitatory, which is why acetylcholine is also released not just at skeletal muscle, but it is also released at a muscle type that has stripes, but happens to have branches on it and a single nucleus. What type of muscle would that be again?
Cardiac.
Cardiac muscle. So this was skeletal. This is cardiac. And so when you sprinkle acetylcholine on cardiac muscle, guess what the effect is?
Inhibitory.
Say again?
Inhibitory?
Inhibitory.
Yeah.
So it makes it fire slower?
Yeah.
Yeah, but wouldn't that make acetylcholine inhibitory?
Yes.
And didn't we just say acetylcholine was excitatory?
Only for skeletal.
Ah, exactly. How? How is that possible? You are 100% absolutely correct. If you take acetylcholine and sprinkle it on a beating heart, that heart beats slower. So on cardiac muscle, it actually has an inhibitory effect. Whereas we know for a fact on skeletal muscle, it has an excitatory effect. So how is that possible? How can acetylcholine be both excitatory and inhibitory?
They have different channels.
Exactly. What makes them excitatory and inhibitory isn't the neurotransmitter itself, it is the channel that it actually binds to. As we know, here on our skeletal muscle, it binds to a cation channel. And that cation channel mostly lets sodium in. All right? That's what makes it excitatory, is because the acetylcholine is binding to a cation channel that is letting sodium in. Whereas it turns out cardiac muscle has a completely different type of receptor. Here, the receptor is actually a chloride channel. So when acetylcholine binds to it, chloride goes in, and that makes the cell more negative. It's inhibitory. So notice whether a neurotransmitter is excitatory or inhibitory isn't actually determined by the neurotransmitter, it is determined by the channel. So it's the two things together, not just the neurotransmitter, but the special type of channel that it binds to. And again, one of the keys that we've talked about is we want this to be modifiable. We want to be able to modify our communication. And there are lots of ways we can do that. In the neuron, we can, this is going to be way too big. There we go. We can make more neurotransmitter, or we can release more neurotransmitter. If we make more neurotransmitter or release more neurotransmitter, we're going to get a bigger signal. We can do things to the synapse. We can move the cells closer together. If they're closer together, more is going to diffuse across. We're going to get more of an effect. Remember, I think some of you got this as an essay question. We could remove enzymes from the neuromuscular junction. Remember, we removed the acetylcholine esterase, and that left the acetylcholine in the synaptic cleft for a longer period of time. It still would eventually diffuse out or be endocytosed, but it stayed in there for a longer period of time. That would give it a bigger effect. And we can change it on the target cell side as well. We can make more receptors. If there are more receptors on the cell for this to be able to bind to, then a smaller amount of neurotransmitter will have a bigger effect. So again, these modifications are dynamic. They're changing. You may not think of it in those terms, right? You may not have ever thought about how modifiable and dynamic the modification of that neurotransmitter is, but you're probably aware of it in one term or another. You think back to the very first time you took a hit off of a cigarette, right? There you were in 3rd grade and your dad had left his cigarette in the ashtray. And so you grabbed a hold of it and took a big puff out of it, right? And what kind of an effect did that have on you?
I started coughing.
You coughed?
Yeah, you coughed. Maybe you made your stomach queasy, right? You're all twitchy as a result of it. That nicotine had a huge effect on your body. And unfortunately for some of you, it may have been a little addictive. So then by the time you were in 5th grade, you were up to 2 cigarettes a day. And by high school, you were at a pack a day, right? Notice when you're smoking a pack a day of cigarettes, does each individual's puff of a cigarette have that exact same effect that it did before?
No.
No, because what happens is you habituate to it. One of the ways you habituate to it is actually the cells that are receiving the stimulus from the nicotine. What they actually start to do is they actually start to remove receptors from the surface, making them less sensitive. So now it takes more of that nicotine to get the same effect, or more of that caffeine to get the same effect, right? That first time you had a shot of espresso, you were running around the house like a crazy person for hours and hours and hours.
Yep.
Now that quad shot of Dutch Brothers barely gets your eyes open in the morning, right?
Unfortunately,
Because of that dynamic changes in these communications, in these synapses.
Is that the same for like medication? So if someone's
Medications,
on,
yeah,
okay.
medications, recreational drugs, toxins, all of these types of things.
So for medication, when people take medicine breaks, what exactly does that help replace those receptors that were once taken off?
Exactly. As you stop taking the medication, the habituation to it starts to go away, and the cell will start to build back those receptors so that when you then use the medication again the next time, you see the effect that you saw the first time.
Is that the same with coffee too? Why people go intermittently without the coffee so it still works?
Yes, I mean, theoretically, I don't know anybody who drinks coffee who has stopped for a period of time for that, but yes. Oh, really? All right, excellent. All right, so yes, the same kind of thing would happen. Anytime you stop taking some type of stimulant like that, like the caffeine, and then you came back to it again, then yes, typically the effect is going to be stronger after wow. Well, you're pregnant though. That's why you didn't have a choice as to what you didn't decide to stop. You had to stop. But yes. So yes, when Audrey goes back to caffeine.
Aubrey's not pregnant.
Wait, you're not the one who's pregnant?
Yeah, I'm not pregnant.
That's Amanda.
Oh, who's pregnant? I know I have.
It was Amanda.
Oh, okay. I'm sorry. The
Yeah.
A. That's the thing, pregnant.
Pregnant too, but I just drink decaf.
All right, then why did you stop?
It was like once the school year ended and I was like, I shouldn't be drinking a pot of coffee and then be tired like an hour and a half later. So, and it worked. So,
There you go. So how long were you off of it for?
like maybe almost 3 months.
Oh, wow.
Oh,
Yeah.
wow.
Yeah. So during that time, when you weren't, and again, it's when you're being inundated by that signal, when there's too much of the signal, the cells start to withdraw their receptors. They get overloaded by it. And so they become less and less sensitive to it. So then, yeah, when you take a break, that brings back the sensitivity. And again, it even something as simple as spicy foods. You eat a lot of spicy foods, and the spicy foods tend to damage your taste buds. So your taste becomes a little less sensitive. Then when you stop eating those spicy foods for a while, your taste buds come back, you become more sensitive to it again. So absolutely. So all of these are examples of these dynamic changes that can take place in how our neurons communicate. So again, number of neurotransmitters released, how long it's in the synaptic cleft, and even the receptors on the other end. All of these are ways we can modify communication, making it more sensitive or less sensitive. Excellent. Last thing, and then of course, like we've talked about, toxins and blockers and things like that can affect communication there as well. Last thing I wanted to do before we take our break and we're done with lecture is to talk about some examples of some of the neurotransmitters. Acetylcholine, as I mentioned, is our most common neurotransmitter in the peripheral nervous system. And again, notice it's excitatory or inhibitory, depending on the type of receptor. So like on skeletal muscle, it's inhibitory, but as we saw on cardiac muscle, it's excitatory, and on cardiac muscle, it's inhibitory. Some other small molecule neurotransmitters are ones that are based on amino acids, including glutamate and gamma aminobutyric acid. And you know how I feel about abbreviations. However, this is one I will give you. You just need to know GABA. You don't need to know gamma aminobutyric acid. The GABA is fine, but glutamate and GABA are important because these are the most common neurotransmitters in the central nervous system.
Do they have a plus and a minus because one's
Glutamate is excitatory and
okay.
GABA is inhibitory, exactly. So glutamate is the most common excitatory neurotransmitter in the central nervous system, and GABA is the most common inhibitory in the central nervous system. Some neurotransmitters are hormones, things like epinephrine and norepinephrine, which may or may not be terms you're aware of, but there is another name for epinephrine. What's the more common term used for epinephrine?
Adrenaline.
There we go. Adrenaline or noradrenaline. What is adrenaline and noradrenaline used for?
Running away from the bear.
Yeah. Stress responses. Absolutely. The adrenaline is part of that fight or flight stress response. Absolutely. To deal with that bear with an axe, right? You either have to fight that bear with an axe or you have to outrun somebody in the room.
Mhm.
All right. Dopamine is another common hormone-based neurotransmitter. Anybody know what dopamine is involved in?
Happiness.
Yeah, absolutely. Reward and happiness. We talked about dopamine earlier, remember, when we were talking about Parkinson's patients and the effects that it has there. But more recently, dopamine has been getting a lot of, a lot of recognition as its association with reward and addiction. Also, a lot of social anxieties are related to low dopamine levels. So the importance of dopamine as a very positive excitatory hormone in the nervous system, central nervous system is really, really important. Speaking of feeling good, we have our endorphins. What kind of things cause endorphins to be produced?
Working out.
Working out can provide endorphins. What else?
Spicy foods?
Not so much spicy foods, but there is a food that can produce endorphins.
So euphoric.
Yeah.
Chocolate? No.
Endorphins, which one? So endorphins are the ones that are, again, working out. It's that high you get from working out. What is the other thing that produces endorphins? Really? Being in love,
Oh,
right?
Yeah.
And what's the food that triggers the exact same reaction in your body
Chocolate.
as being in love? There you go.
Yes.
Chocolate, exactly. Working out, being in love, and chocolate. There you go. All of those release endorphins in your body, right? We've been dealing with that while we've been here stuck at home, right? Being stuck at home, we may not be necessarily with our loved ones anymore. So what have we been doing? You're working out or eating chocolate, right? As they said, when this quarantine began, you were either going to become a hunk, a chunk, or a drunk, right? And if you worked really hard, you could be two of the three. So yeah, so there you go. All of those to make up for those endorphins that we've been missing from all of our social interactions. These are all ones that you may be familiar with. Substance P, you probably haven't heard of before, but with a name like Substance P, any idea what it might be involved with, what that P might stand for?
Pain pathways.
Pain, exactly. Substance P is one of the primary neurotransmitters involved in our pain pathways. Obviously a very important, but not nearly as studied as it should be pathway. We don't understand our pain pathways quite as well as we should, but we do know that substance P plays an important role in it. And not surprisingly, with over 100 different things being identified as neurotransmitters, there can be some very interesting things. ATP can actually act as a neurotransmitter. Some gases like nitric oxide. Notice not nitrous oxide, that's what you get at that dentist's office or that rave on Friday night. But nitric oxide plays an important role in changing the diameter of our smooth muscle, especially of our blood vessels, helping to regulate where blood goes through our body. And even things like calcium and some other things can be as well. Your book's got a nice table recognizing many of the different neurotransmitters. You don't have to memorize all of those, but at least be familiar with some of these more common ones that we have talked about. All right. Questions on that?
Are we allowed to use, like, for example, number 3, like hormones or adrenaline as, are they interchangeable? Okay.
Adrenaline and epinephrine are interchangeable the same way that posterior and dorsal are interchangeable terms. So yeah. And again,
And the biogenic amines, can we say hormones, or do you want us to use
yeah,
it
no,
there
same
too?
hormones would be
Okay.
absolutely. Right. And again, really, I think it's interesting. Everybody thinks of adrenaline and in the body, noradrenaline, but like, as I mentioned, my daughter has a nut allergy. So what do we carry with us everywhere we go?
EpiPen.
An EpiPen, right? That EpiPen contains epinephrine.
That's
Right?
super expensive.
Yes, absolutely. So my daughter has her EpiPens that she carries with her everywhere she goes now, because she's, being a teenager, she's more responsible. We used to carry them for her all the time. And they now have these nice compact flat ones that we thought would be really, really nice for her, to be easier for her to carry in her purse and carry around. So we contacted our doctor about getting some of those, and because they're a specific brand, they're not carried by, they're not covered. So, but we could purchase them if we wanted, and they're $5,000 a pen for these flat ones. So she needs a bigger purse. She's using the old ones that we have. Yes. So yeah, it is pretty ridiculous.
Don't they expire too? Like, don't they have a short shelf life?
Yes, the adrenaline does not expire. So back in olden days, you would get a vial and a hypodermic needle, and basically it would last forever. The problem with the auto injectors is the mechanism that causes the injection will, in time, can start to fail. So it isn't that the medicine goes bad. It's more of an issue of the mechanism that allows it to auto-inject can become less reliable. And so because of that, they do have an expiration date. They can, if it injects the medicine past that time, it still works. And most of the time it is. It's just that there's a success rate that starts to decrease where, you know, from a liability standpoint, they're not comfortable with. So they do put, expiration dates on it. But the whole point of that EpiPen, again, if she were to come in contact with that nut protein, she could have basically two main effects. You get a vasodilation, which causes a drop in blood pressure, making it hard for her to get blood to her brain, and thickness of the mucous membrane, making it harder for her to breathe. Can't get the oxygen. So what happens when you get that adrenaline, when you hit her with that EpiPen? Increases blood pressure, helping to get blood to the brain, loosens the smooth muscle of her airways, hopefully helping her to get the air into her lungs, and hopefully helping her to survive that allergic reaction. So yeah, so again, it's that stress hormone, that stress response. All right, questions on that? All right, that is everything I wanted to cover for lecture today. So let's do this. Let's get rid of that. Let's get rid of that. Where did we leave off here? We left off here. All right. As I mentioned, we have done good with lecture, but where we're a little behind now is on the anatomy. So what we're going to do after the break is come back and pick up where we left off, talking about the anatomy of the lobe. The lobes, the regions or areas, and their functions. We've gotten through two of the lobes, which means we have 2.5 to go. So let's go ahead and take our first break. We went a little long. It's 9:27. Let's go ahead and I'll give you guys a 20-minute break because we went so much longer. So let's restart.
Thank you.
At 9:47, and we'll start the recording at that point. All right, so that gives you guys a little bit more time to stretch, get something to eat, biological break, and I will see you back here in 20 minutes. All right, bye. Any questions before we get started? All righty, excellent.
I actually had a quick question. Sorry.
Yes.
So I believe we went over the general interpretive area last time, which was on the left side, but you also mentioned something being on the right side. I just forget what it was and if we're responsible for that.
Spatial awareness. Remember
Okay.
the person who forgets about the contralateral side of the world, including their
Okay.
body? Yep.
Thank you.
Absolutely. And actually, that is the one advantage of having split this in half, is we get a chance to do a quick review. So again, I've got the telencephalon located here. And I have two structures that we have identified here. Identify this structure or groove. Now, my simple illustration, what does this line here represent? I guess it's a good thing we're doing a review.
The
Central
Central Sulcus.
There you go. Excellent. And what does this line represent?
The lateral sulcus.
Excellent. That helps to divide, these two help to divide our cerebrum into 4.5 lobes. And what was this lobe up here?
Frontal lobe.
How many areas or regions are we responsible for in the frontal lobe?
And I know 3 of them.
3 of them. Excellent. Right. One of them was the region in the very front of the frontal lobe. And what was this area called?
The prefrontal cortex.
And what was its function?
Intellect, personality, and reasoning.
Excellent. We had this region here that kind of had these three parallel gyri on them. And what was this area or region called?
Speech.
Sarah's already beat me to it. Yep. Broca's, or also speech area would be acceptable. And of course, what did that do?
Produce speech.
Yeah, produce speech, absolutely. And remember, this was one of those lateralized areas where most people is found in the left hemisphere.
So then for these two, like for example, we have on the right side, it's what, like compute, I don't remember. It's
Mathematical skills, yeah.
Right. So when you talk about like the right and the left, are we responsible for both of those? Like if you were to ask us on the right side of the brain, okay, what is this area? Because technically it wouldn't still be like speech area, right? It would
would be some kind of computational area, yes.
Right.
So
okay.
You're right. If I showed you the right hemisphere and I asked you for the function, then yes, or the the common function found on this hemisphere. My guess would be that the question would help to emphasize that this was a lateralized area. Like on this hemisphere, what is the likely function of this if I were to show the opposite side?
Oh, okay.
Yeah.
Okay, thank you.
All right. Now, one more quick thing. There is the same way there are these three parallel gyri located here. There is also a gyrus that is here to the anterior side of the central sulcus. What is this structure called?
Pre-central sulcus or gyrus?
Excellent. This structure is the precentral gyrus. However, on the precentral gyrus is a region or area. What is the name of the region or area? There we go. Excellent. Thank you, Sarah. Primary motor cortex. And what is its function? I know I didn't write them out for the other ones, but I want to make sure we do it for this one because we know the difference between the structure and the area. What is the function of the primary motor cortex?
It voluntary controls the contralateral skeletal muscle.
Excellent. Notice there is a lot of vocabulary in that one, so that's another good reason to write that out. Voluntary control, contralateral skeletal muscle. So the left side basically allows you to move the right side of your body. Excellent. That was our frontal lobe. Then behind the central sulcus, what lobe did we have here?
Parietal.
Excellent. And how many areas or regions in the parietal lobe were we responsible for?
2.
2. One of them was here at the bottom of the parietal lobe and may have snuck onto the temporal lobe a little bit. What was this area known as?
Wernicke's.
Wernicke's area. Or what was the other name? Anyone remember?
The general interpretive.
There you go. Usually the Bob name is the one that's harder to remember, but I think in this case, Wernicke's, I think, sticks out more than general interpretive does for people. But either way, what does Wernicke's area or the general interpretive area do for us?
Interpret like speech and produce meaningful speech.
Excellent. I love that. Interpret speech and produce meaningful speech. Remember, we saw that individual that had the aphasia, that Wernicke's aphasia, where again, if he was sitting behind you at a Starbucks and you weren't paying attention, you wouldn't think anything about the conversation taking place. His speech had all the normal inflections and cadence and speed that you expect from normal speech, but it's not till you start listening to the words that he's saying that you hear and realize that he's got extra syllables, or the words don't make sense together, or he's not producing meaningful speech. Excellent. Secondly, once again, there is a structure behind the central sulcus. What was that structure? There we go, thank you. Post-central gyrus. Excellent. And on that structure, again, is an area of cortex that has a specific function. What is that area or region called? Thank you. Primary somatosensory cortex. And what did that primary somatosensory cortex do?
It received the general sensory information of the contralateral side of the body.
Yeah, basically touch from contralateral side. Of the body. Excellent. So again, on that post-central gyrus, we have that primary somatosensory cortex. Excellent. And let's move this here. Our third lobe is down here below the lateral sulcus. And what lobe was this?
Temporal.
Excellent. And our last lobe was here to the back. What was this last lobe here in the back?
Yeah, occipital.
Excellent. So those were our four main lobes. We'd identified the regions of two of them. So let's finish the last two, starting first with the temporal. For the temporal lobe, how many areas or regions are you responsible for? Two. Absolutely. And notice my drawing doesn't do a good job of showing this, but if you notice, the central sulcus basically comes almost all the way down to touch the lateral sulcus. And that's good because basically what it points at is an area located right here. And this area, because it's so small, I'm not going to have a chance to write it here, so I'll write it over here, is what we call, let's number it. Is the primary auditory cortex. What do you think the primary auditory cortex does?
In charge of listening?
Receive sound information, but let's be a little bit more specific. If primary motor cortex controls the contralateral side, if primary somatosensory receives touch from the contralateral side, what do you think the primary auditory cortex does?
Receive sound from the contralateral side.
Exactly. Or really, if we wanted to say, from the contralateral ear, right? So information coming into the right ear goes to the left side of my brain. Now, one of the important things to remember is with the primary auditory, really primary anything. When we talk about these primary areas, basically, or sensory primary areas, they receive the raw data. They receive the sound. So if this area was damaged, what would happen?
We can't receive any sound.
Yeah, basically, if you damage it here on the right, you would be deaf in your left ear. No information coming in your left ear would reach the brain. And so you just wouldn't perceive any sound from that ear. However, once we receive that information, as we know, our brain needs to be able to process it. Our brain needs to be able to make sense of it. And so surrounding, and I'm exaggerating this a little bit for effect, but surrounding the primary auditory cortex is our association. And association cortex, their job is to make or give meaning. Let's say it that way. Their job is to make sense or give meaning to the raw data. Right? Primary is where we receive the sound. However, our association cortex is what tells us it's an oscillating sound that is oscillating between two frequencies, and the amplitude is increasing, meaning that I'm hearing a noise that is getting louder, going back and forth between two frequencies. Wee-oo, 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 wee-oo. And that is where it makes sense. Oh, hey, that is a siren. Maybe I should get out of the middle of the street. So the primary cortex receives the raw data, and association cortex is what makes sense of it. And that's what these two areas are. Notice I've done an amazing job of drawing them here on our pretty picture, but if we come to here, we can see the same thing on our illustration here. Notice, again, follow the central sulcus down, and it pretty much points right at the primary auditory cortex. And then the association auditory cortex is this area that surrounds it. All right. Questions on that?
How do you suggest we study for this? Like for the muscles?
Very
you're gonna give us
carefully.
Okay.
Like I said, I think, again, this information is a little less intuitive because the part of our brain that is involved in hearing doesn't look like an ear. The part that, allows us to feel with our hands doesn't look like a hand. So notice one of the things we've been doing is trying to break it up into little, small, easily digestible pieces. Not only are we just looking at the cerebrum, but we're looking at each individual lobe. Notice for the temporal lobe, there's only two things you need to know. For the parietal lobe, there's only two things that you need to know. Heck, for the frontal lobe, there's only three. So try to break it up into smaller, more easily digestible pieces. Yes. And also contralateral. Excellent. Yes, this is also contralateral. To more easily digestible pieces. So really, that's how I think works best. We've been trying to break it up into small pieces, and each of those small pieces, right? Knowing two things in the temporal lobe shouldn't be that hard. All right, so it's just a matter of breaking it into these smaller, more easily digestible pieces, and that'll hopefully help to make more sense of it. Okay.
For the association auditory cortex, is it receiving or giving meaning to the sound information?
Yes.
For the contralateral? Okay.
Yeah,
Not
to
just
the
giving
raw,
meaning
to the raw,
to the...
yeah, to the raw data of the sound.
Okay.
Or let's say the raw data from the primary area. All right. So the primary area gets the raw data, gives it to the association area to make sense of it. Notice I've made a point of emphasizing the primary area and the association area for auditory, because where else is there primary and association areas? If only I had my anatomy listed in front of me. Yeah, the occipital lobe, absolutely. Way back here in the back of the brain is Brodmann's area 17. And you don't have to memorize that number 17, but this area 17 is our primary visual cortex. And guess what the primary visual cortex does?
Receives raw data.
From where?
From your eyes.
No.
Okay, well, I don't
close.
know.
Oh, okay, obviously, yes. It's visual information from the eyes, but let's be more specific.
From the retina.
Okay,
I don't know.
but let's think about this. Touch from my right hand goes where?
To our left.
Right.
Right.
Touch, hearing from my right ear goes where?
To our left.
Yeah. So Sarah pointed out the left eye or the right eye. So let's do that. Close your left eye or cover your left eye. When you cover your left eye and look ahead, do you only see the right side of the world?
No.
No. So notice your eyeballs have a problem. Unlike touch, which all goes to the opposite side, unlike hearing, which all goes to the opposite side, what we want to receive is from the raw data from the contralateral side of the world, not of the eye, of the world. We want everything from the nose over here on the right to go to the left. We want everything from the nose over here on the left to go to the right. And the problem with that is your eyeballs receive both. They receive information from both the right side of the world and the left side of the world. Notice your brain or your nervous system has figured out a clever way to deal with this. Here are my two eyeballs. Notice for each of my eyeballs, they both receive some information from the left-hand side of the world. The stuff from the left eye and the left-hand side of the world, that information we want to cross to come to the right side of the brain. But the stuff over here on the right is already on the right. So we want it to stay on this side. And the same thing is true for the right. Notice stuff from the right eyeball and the right side, we want to cross to the left. And stuff that's already on the left, we want to stay on the left. We have this special area known as the optic chiasm. And this optic chiasm is where we get a partial crossing of information. And so instead of being eye-specific information, which is what we have in the eyes, right? If I close my left eye, I don't see any of the information from the left-hand side. However, if I were to damage my primary visual cortex in the left hemisphere, I would see nothing from the right-hand side of the world. No matter which eye I used, this back here in the brain is now side-specific and specifically contralateral. So notice our primary visual cortex receives the raw data from the contralateral side of the world. It's hard to imagine what that's like. It's easy to close one eye or cover one eye and see what it would be like to have just one eye. but it's really hard to, in our mind, visualize what it'd be like to lose all sight to the right side of your body, no matter which eye was open. No matter which eye you were using, you would see nothing on the right side of the world. That would be what would happen if you damaged area 17, your primary visual cortex. You would be blind. You would perceive no light from the whole right-hand side of the world. It's kind of trippy to think of, but that's exactly what would happen. And it's because of this magical thing called the optic chiasm, where some, but not all of the information crosses. Notice all of my sensory crosses, all of my hearing crosses, but not all of my vision. Both sides of the brain receive information from both the left and the right eyes, which is important. That gives us binocular vision. That's what allows you to thread a needle. Having that binocular vision, being able to see both the right and the left sides of the world. So, raw data from the contralateral side of the world. And of course, once we get that raw data, guess what we have to do with it?
Give meaning to it.
We must process that raw data. And how do we give meaning to that raw data?
The visual association area.
Exactly. Or areas. Some of it is pretty basic. Again, you're not responsible for these numbers, but not surprisingly, right next to area 17 is area 18. As I mentioned, if you damage area 17, what do you see?
Only one half of the world.
Yeah, well, if you damaged area 17 for that side, you'd see nothing, right? If you damaged area 18, you wouldn't see color. Area 18 is what processes color, makes sense of color. Remember, I fully admitted that I am biased because I'm a visual neuroanatomist by trade. But as I also mentioned, over 40% of your cortex is involved in processing visual information. In fact, there are two visual processing pathways. One is what is known as the what pathway, and the other is what is known as the where pathway. Guess what the what pathway is involved with processing? Yeah, what an object is. This what pathway actually travels into the temporal lobe. And its job is to tell us what something is. Make sense of the visual information that we are learning. And the where pathway goes up into the parietal lobe. Is responsible for telling us where something is in space and how it is moving. Doesn't matter what the object is. It could be a red dot. It could be your grandmother on a skateboard. Doesn't care what the object is. It just tells us where it is and how it's moving. Again, I think sometimes it's easier to understand what these areas do when we see what happens when they're injured. I'm going to give you two names of two areas. You are not responsible for the names of these two areas. I just want to mention them so that you can see and understand. In our where pathway, there is a region called MST. And MST is responsible for optic flow. What I mean by optic flow is if you get up and walk forward in space, Notice as I'm coming forward in space, the world around me is expanding. If I rotate in my chair, as I move, the world moves by me in the other direction. So I can not only tell where something is in space, but how it's moving by this optic flow. There are individuals who have damage to their MST area, which means they can't observe optic flow. Well, what does that mean? What is their word like? Well, remember how we talked about using that nitrous oxide at the raid on Friday night? Well, you're at that rave on Friday night. And of course, any good rave is going to have a strobe light. If I'm on one side of the rave and you're on the other side, and I'm waving to you, and that strobe light is going off, do you actually see my hand going back and forth as I'm waving to you across that strobe light?
No.
You see my hand here, and then suddenly it's here, and then suddenly it's here, and then suddenly it's there. It's like a bunch of still images. That is exactly what it's like if you have damage to your MST area. You do not see movement. It's like constantly living in a strobe light. Can you drive a car if you have damage to your MST area? No, absolutely
No.
not. Right? Can you pour a cup of coffee if you have damage to your MST area?
Yes.
Well, I mean, static images here, and then suddenly it's overflowing, right? You wouldn't think that they'd be able to do it. Now, in all practicality, do people with MST pour themselves cups of coffee in the morning?
Yes.
Yeah, how do they do that?
They're like
Very
this.
slowly. They have a Keurig.
Nope. Aubrey. Awesome. Absolutely. That would definitely be one way to do it. Although Aubrey hit the other cue. You put your thumb in the mug. You put your thumb in the mug and you keep pouring till the coffee comes up to your thumb. You know, if you think about it, you could do that with your eyes closed if you did it that way. So yeah, so in practicality, they're able to do it, but they can't do it just by watching. What pathways even more interesting? This allows us to make sense of the images that we're seeing. There is an area in the what pathway called the Intra-Temporal. So it is called IT. And it actually allows us to make sense of the images that we're seeing. There is a very famous individual who has damage to the IT region who has been studied for many, many years, and there's been many books written about him. And one of the classic cases with him is he was shown a glove, right? He was able to describe the glove, that it had a central region, and then it had these 4 extensions that extended out in one direction and a 5th that extended out in the other. He was able to identify that it had yellow and blue alternating stripes on it. He could tell that it looked like it was knit. He could describe the thing perfectly. But then they asked him what it was, and he sat and stared at it for a while, and looking at the extensions going out, he's all, oh, that must be a coin purse, right? One of these is for the pennies, one for the nickel, one for the dimes, one for the quarters, and so on and so forth. He could clearly see the image, but he couldn't put a label to it. Now, once you hand it to him and he can add that tactile sensation, he instantly knew it was a glove. So he can see things, but he can't put labels on them. He happens to be married. And if his wife is standing still, he does not recognize her. The second she talks, he can recognize her. Sometimes when she walks, he can recognize her gait and he can recognize her that way. But at one point during an interview, his wife was sitting there. They asked him what was sitting next to him, and he said a hat. Right. And like I said, he's a very famous case, and there's actually many books written about him. One of them is The Man Who Mistook His Wife for a Hat. And so that was one of the impetus for one of the books that was written, and a very famous case of this guy with this very precise deficiency. And it kind of shows you this MST, this IT deficiencies show you just how precise. We go from just seeing light or just seeing color to very precise pieces of information. A tremendous amount of processing of that raw information to make sense of it.
Is that why everyone's always like, be careful, like if you slip or hit the back of your head or cover the back of your head?
So you are correct. When you fall or someone punches you in the head and you see stars, that seeing stars is typically from the back of the brain hitting your skull, depressing that primary visual cortex. When you depress that, it stimulates it and you see flashing white lights. So that stars that people see when they take a blow to a head is because of trauma to that primary visual cortex.
So for the visual association area, are these two pathways technically part of it?
Yes, they are the main part of it, absolutely.
Okay.
Yeah. So not only are they making sense of the information, again, raw association areas make sense of the raw data, they're making sense of it in very precise ways, identifying where something is in space and what something is.
So if he asks us for the function, it's processing the raw data from the primary visual area. But then if he asks us the pathways, one of them is the where and one of them is the
What?
pathway or what? Yeah.
Where and what?
Okay.
All right. So there you go. Questions on that? So those are our 4 lobes. However, I have not so subtly been emphasizing 4.5 lobes, as I keep mentioning the lobes of the cerebrum. And the reason for that is if you were to actually take a couple of clamps and you were to peel open the lateral sulcus, what you see inside of the lateral sulcus is a big, large chunk of cortex. Notice another way to see this is in the in the coronal section view. Notice in this coronal section view, we see here in the lateral sulcus, there is this big, huge piece of gray matter. And this big, huge piece of gray matter is what is known as the insula. And there are some anatomists who think of the insula as a 5th lobe. Honestly, I don't care whether you call it a 5th lobe or don't call it a 5th lobe. You just need to know that it's the insula. And you need to know what the insula does. The insula basically has kind of 2 main functions. One of the main functions of the insula is it plays a huge role in processing desire, fear, anger, and addiction. We were joking before about smoking that cigarette at 3 years of age or 3rd grade. 3 years of age would be outrageous. 3rd grade, much more appropriate. But there are plenty of people who try a cigarette, think it's gross, and never have it again. But there are plenty of other people who have that first cigarette and have to have more. And it's not just cigarettes, it's drinking, it's gambling. Gambling can be an addiction. And what's really interesting about this is that they're finding, again, one of the huge things that has helped us to understand the processing of the brain is a process called a functional magnetic resonance imaging machine, or what they call an Fmri. And basically what this is able to do is it measures blood flow to parts of the brain. So they're able to put you in this big, huge magnet, and they have you looking at pictures. And when you look at pictures, your visual cortex in the back of your brain lights up. Or they play music and your auditory cortex lights up. Or they touch your hand and your primary somatosensory cortex, where your hand is, lights up. what they will do is they will put individuals in this fMRI and they will flip a coin and they'll have them guess whether it's heads or tails. And they do that in normal people, and they do that to gambling addicts. And what they find is when they do that to gambling addicts, there is a huge increase in activity in the insula that we don't see in those people who are not addicted to gambling. Narcotics, nicotine, sex. For those people who have addictions, those stimuli cause a much larger response in the insula. This is a huge finding, because if we can block the activity in the insula, we may be able to make it easier for people to break these addictions. If they're not getting the same stimulus, right, the same way that a normal person doesn't get that intense stimulus there in the insula, that may help them to break those addictions. So this is a really, really interesting field of study. The other big key to the insula is the cortex, right? The cerebral cortex, the gray matter, that, how do we want to say this? Oversees a region of the brain known as the limbic system. If you notice, as we saw before, I'll use blue for this. Here deep in the cerebrum are all these nuclei. Remember, these nuclei are, I'm gonna have to make this smaller now. Are like ganglia in the central nervous system. They're clusters of cell bodies in the central nervous system that are deep away from the cortex. But they're still involved in processing information. I've actually written down here how I want to describe what the limbic system does, because I want you to have this precisely. The limbic system is this gray matter between the cerebrum and the diencephalon, the part of the brainstem, and it is a functional group that is responsible for emotions, memory storage and retrieval, and here's the part that I wrote down because this is the part that I wanted to make sure that you got: linking conscious thought and unconscious function. What do I mean by that? What do I mean by linking conscious thought and unconscious function?
Is that kind of like interpreting your dreams, sort of?
That could be a part of it, absolutely. If you're having if your loved one sitting next to you is having a bad dream, are you aware of it?
Yeah.
Yeah, depending. Their breathing may increase, right? They may be twitching more, they may be moaning or something like that, becoming more twitchy. And do they even need to be asleep for that? Right? You come stumbling home drunk, 3 o'clock in the morning on a Wednesday, fumbling with the keys to get inside the door, and your significant other has to come and unlock the door for you. And without them saying a word, can you tell what they're feeling and thinking about you at that moment in time?
Yeah.
Right? That blood vessel pulsing on their forehead, the shaking of their hands as they're sitting there and tightly, right? The white or the reddishness of the face, right? All those things clearly tell you that they're madly in love with you and want you to take them right there, right? No, but that ability where what we're thinking affects our body. When we're sad, when we're angry, when we're aroused, when we're scared. If you think about it, that's the whole point of a lie detector. The lie detector, they hook you up, they're measuring your blood pressure, they're measuring your heart rate, they're measuring your skin conductivity. Because, right, they're asking you all these casual questions. What is your middle name? Do you like puppies? Where were you on the night of the 4th? Right? And if you were doing something wrong on the night of the 4th, does your heart beat faster? Does your blood pressure go up? Do you become a little sweaty? Do you become more twitchy in your chairs? Yeah,
Yes.
they're measuring all those things because what you're thinking has a physical effect on your body. And this limbic system is the area that is responsible for that. We can see that when we take a look at some of the structures that are found in here. Notice these are some cool pictures from your textbook where rather than a coronal section, we see a partial sagittal section, and then where we see kind of the entire structures in here. And we can see some of the structures that are here in our limbic system. Let's start first, let's go back to this picture with this purple region right here. So notice there's that purple region right here, which when we look at this view, is this purple region right there. This almond-shaped region is known as the amygdala or the amygdaloid body. The amygdala is a structure that plays an important role in our memory and our emotional reactions, especially anger and fear. As I mentioned, without even saying a word, the facial expression of your loved one can tell you what they're thinking about right there, whether they're happy, they're sad, they're aroused, they're angry, they're bored. We're very good at reading facial expressions because we're very social. But it turns out monkeys are also very social. And especially like Rhesus macaque, have a very rigid social hierarchy. And part of that social hierarchy is facial expressions. They have two main facial expressions. They have an aggressive facial expression, and they have a submissive facial expression. And monkeys are really, really good at recognizing this. We know this because we can strap them in a chair, and we can show them an image, and we can give them 2 buttons. And if they see an angry face, they push the first button, and if they see a submissive face, they push the second button. And when they do this activity, they get rewarded with a little bit of juice, and they love juice. So they will sit there for hours, if you'd let them, doing this activity, just recognizing angry and submissive faces, and they're really excellent at it, unless their amygdala has been damaged. If their amygdala has been damaged, then they lose that ability to be able to recognize the difference between submissive and angry faces. Now, hopefully you've never been strapped to a chair, even if you're going to get juice, and had to look at angry and sad faces. But you may know the amygdala by the name that we call it, or at least we used to call it here in our house. The amygdala is the worry monster, right? My daughter, when she was very young, had a very robust amygdala. The amygdala's job is to keep you alive, right? Because again, if you think from an evolutionary standpoint, we used to live in trees, and bears with axes roamed the countryside. So every time you heard a board creak or a limb snap, it could be that bear with an axe coming to get you, right? So my poor little daughter, sitting there in her bed late at night, hears a car door close outside, and she panics, right? Or she hears a creaking of the heater getting ready to turn on, and she freaks out, thinking it's that bear with the axe, right, coming in to get her, right? So that worry monster is that job of the amygdala. It over-exaggerates those fears, or can over-exaggerate that fear response in that protective nature, right? So you got to get out of that amygdala, get out of that knuckle of your thumb, if you think of that glove, and get into your prefrontal cortex, right? So there's all those tools you use to calm your mind, to get to sleep, to get rid of the worry monster, right? Count back from 300 by 3s, right? Or you think of an animal for every letter of the alphabet, starting with A and working your way down, right? There are all these tools that can be used to get out of that worry monster and get into that prefrontal lobe so that we're able to negate the effect of that worry monster and calm the brain and get to sleep. All right. Questions on that? All right. Notice that last picture didn't show it, but here we see off to the side, we see this fuzzy red thing. And I hate this picture from your book that shows the fuzzy red thing because it just shows it as this amorphic blob. But that's not actually what it is. What it is this area that has kind of a large body, and then it has this curly Q tail to it, which is actually where it gets its name. This structure is known as the hippocampus. And anyone know what hippocampus refers to? You ever heard that word before?
Hippopotamus.
Close. Hippopotamus is similar to that. It is something water-based. A hippocampus is the Latin term for seahorse. And that's what this looks like. You got the body of the seahorse and then the curled tail of the seahorse. That is where the hippocampus gets its name. The hippocampus' job is to convert short-term memory into long-term memory. What does that mean? All right, I'm going to identify three things for you. Boat, ball, cat. What are the three things that I just said to you?
Boat, ball, cat.
There you go. You just used your short-term memory, your short-term working memory, right? However, if I then gave you a list of multiple choice, not multiple choice, mathematic questions, multiplicative questions, right? What's 5 plus 8?
13.
There you go. 2 times 2.
4.
10 minus 3.
7.
1 plus 1.
2.
What were the three things that I said earlier?
Boat, ball, cat.
There you go. Exactly. Notice you were able to store those three pieces of information into your long-term memory so that you were able to process those mathematical questions I gave you and then recall the three items that I gave you earlier. Someone who has damage to the hippocampus can't do that. There is an incredibly famous case of an individual who's only known as HM, who, gosh, over 30 years ago was camping. And as he was camping, it started to rain. So he grabbed all of his camping equipment, put it into his station wagon, and decided to sleep with a station wagon. And included in those things that he put in the station wagon with himself was his kerosene stove.
Oh, no.
He ended up getting carbon monoxide poisoning and got bilateral damage to his hippocampus. And he has not been able to maintain any long-term memory information since then. It's been over 30 years now. And for at least the first 25, one of the primary instructors that was working at him was actually at UC Davis and worked with him for over 25 years. And every morning when he walked into his hospital room, he had to introduce himself to him for the first time. He leaves and goes to lunch and comes back, and guess what he has to do?
Introduce himself.
He has to introduce himself to him again. Absolutely. He has no ability to convert short-term memory into long-term memory. He can remember all the things that happened before, right? And again, even at the time, how well do you remember things that happened 10 years ago? Yeah, not that great. Well. So again, there is some degradation to his memory, and not surprisingly, 30 years later, he has trouble remembering what he had for lunch the day before this injury occurred. Things like, not important things like that, but there are still some basic information. But he hasn't really been able to produce new conscious long-term memories. Studying him has allowed us to learn a tremendous amount about memory, because not all memory is the same. For instance, people always talk about muscle memory. Once you learn to ride a bike, you always know how to ride a bike. Well, there's actually something to that. Not surprisingly, HM has a tremendous amount of time on his hands. So one of the things he does is play video games. So you could walk in after lunch. Of course, after you introduce yourself to him again, you can show him a video game. Have you ever played this video game before? And he will swear on a stack of Bibles he's never played that video game before. You then put it into the machine, you hand him the remote control, and he'll get the top score. The whole time swearing he's never played the video game before. All right, that ability, that muscle ability, that memory of the muscle ability is obviously something that is kept different from the cognitive information that he's able to access. Imagine what it's like waking up in the morning. You go to shave. And when you go to shave, is the person looking back at you in the mirror the same person you expect to see?
No.
No. Now, this is a little tricky. He is aware that something has happened, and he is aware that time has passed. But not only was his hippocampus damaged, but his amygdala was damaged as well. So whereas someone else may be barely frightened or really angry to see this old man staring back at him, his emotions are much more muted. Again, there may be others that have had hippocampal problems and removed that way, but I believe HM was because of carbon monoxide poisoning. But again, he's certainly not the only one who's had this issue. So there are other people like that as well. Alzheimer's is a different type of disorder where the cause of Alzheimer's is not fully understood, but it is a degenerative breakdown of memory. Again, newer memories break down more easily because they're not as infirmed, but what actually causes that is not fully understood that way. Many of you may be familiar with people with hippocampal damage because they're two very different but both equally excellent movies that have been made about individuals with hippocampal damage. And of course, what are those two movies? One's a comedy. What's the comedy? Come on, I know someone knows it.
Isn't it? It's the one with the boat.
Exactly. There you go. Brooks got it. 50 First Dates. Absolutely, right. Drew Barrymore and Adam Sandler. Excellent, excellent. One is a psychological thriller. Does anybody, that one's less well remembered, but equally excellent. Anybody remember that? Police officer, a bad guy sneaks into his house, kills his wife, injures him. He ends up having hippocampal damage, but being an ex-cop, he wants to get revenge and find and kill the person who murdered his wife, but has no ability to produce short-term into long-term memory. So he has to get information tattooed on his body, and he takes a bunch of pictures and writes a lot of notes. Memento with Guy Pearce. Excellent, amazing movie. What's really cool about that movie is you see it from his point of view. So with that staticness of the short-term memory and the no ability to produce long-term memories. So it's an excellent movie. If you haven't seen it, I highly recommend it after the semester. But definitely, again, gives you that sense of what the hippocampus does. All right. While we're here, let's look at a structure we've already talked about before. If you remember, up here in the cribiform plate, we talked about that olfactory bulb and the olfactory tract. Remember, we talked about this is what provides the nerves that give us our sense of smell. Talked about my sister's boyfriend who bounced down the stairs and tore his office cribiform plate. Well, if you look closely here, you can see again that olfactory bulb that carries the neurons and the cell bodies, and then the olfactory tract, which are the axons that carry that information straight back to the limbic system. You may not have thought of it in these terms, but I know you're aware of the fact that your sense of smell is the one that is most closely associated with emotion and memory. Because if you're like me and you had that really bad breakup, you may spend some times when you're feeling melancholy that you'll listen to his old messages on the machine so you can hear his voice. Or you'll look at those pictures of that time that you spent together at Niagara Falls. But when you're really, really feeling sad like I do, you've got that shirt of his in the Ziploc bag that you open up just so you can get that little bit of a smell of him, and it brings all the emotions and all the feelings back, right? Our sense of smell is so closely associated with memory, right? Or maybe it's that smell of the baked bread or the apple pie that reminds you of that summer that you spent at grandma's house, you know, swimming in the pond and swinging on the tire rope and all of those kind of things. Our sense of smell is really the only sense that goes directly to the limbic system. So it is the one that is most closely associated with our emotions and with our memory. They're the ones that are able to trigger the strongest emotions and strongest memories because they come straight back to the limbic system. This is important because one of the things they communicate with is this little red fella right here, known as the mammillary body. Now that's weird. Dr. Slutsky is talking about a mammillary body, but I don't see it in the limbic system list. So why is he talking about it? Is it maybe on your list someplace else?
Yeah.
Where?
The cephalic feature.
Diencephalon, absolutely. It is a diencephalic structure. This mammillary body's job is taste or smell aversion. Right? If a sheep out in the grazing out in the pasture eats some red berries and gets sick the next day, is it likely to eat those red berries again? No. And does the same thing work for you as well? Absolutely. It doesn't even have to be the trigger. My wife used to, when she was down in Southern California, there was a restaurant we used to go to all the time that had this amazing black pepper ranch dressing that she would get on the salad every time we were there. She absolutely loved it. And then she got pregnant. And we went there one night for dinner. And then the next morning, she woke up with really, really bad morning sickness, throwing up all over the place, including throwing up some of that old, black pepper ranch dressing. And did she ever go back to that restaurant and eat that black pepper ranch dressing ever again?
Nope.
No, absolutely not. Right? Or maybe, again, back in 5th grade, you drank a whole bottle of Jose Gold tequila, right? And to this day, even the smell of it makes you nauseous, right?
Yes.
So, exactly. So that taste and smell aversion is what that mammillary body does. It associates something, the sense of taste, the sense of smell of something that made you sick, or at least was related to you being sick. And notice the reason we're talking about it now is obviously this mammillary body has to get that smell information from the olfactory lobe, the olfactory bulb and tract, but it needs to connect it to our memory. And if you notice, there is this axon structure, this bundle of axons that carries information from the mammillary body to our hippocampus. And that structure is known as the fornix. So the fornix, which is on your limbic system list, is a bundle of axons that connects the mammillary body to the hippocampus. Notice we see it nicely in this weird pulled out section where we can see the entire limbic system. But notice if we look up here in our sagittal section, we see it right here. And in fact, if we go to this picture here, we see that fornix as well. that white matter structure right here, sitting above the thalamus. And notice this is the mammillary body right there. There we can see these two structures.
So for the limbic system, we need to know the structures and the function.
Yeah, although the fornix doesn't really have a function because it's an axon, it's a bundle of axons, it's a track. So basically it's just a structure that has to be recognized.
For the ones that we do need to know the functions for.
Exactly.
Okay.
Areas and regions and stuff like that, or the nuclei that we've been talking about. Notice the fornix is not the only white matter structure we're responsible for. There is the largest white matter structure we have here in the limbic system is this big comma-shaped structure known as the corpus callosum. Notice we can also see the corpus callosum. Oh, there's the fornix again. See that really nicely there. And the corpus callosum here. But what I want is, no, not that. Let's go here first. There we go. Here. This big white matter structure going from one hemisphere to the other. In fact, it is the largest commissural white matter structure. Remember, commissural just means it carries information from one hemisphere to another. Is the corpus callosum. You saw how huge it is. So its job is to carry information from one side of the brain to the other side of the brain. Like I said, even if you're doing something as simple as clapping, do we need to coordinate what's going on from the two sides? Yeah, absolutely. Now, someone mentioned epilepsy earlier. Epilepsy is a condition where basically a cluster of cells in one part of the cortex becomes super active, producing a huge wave of neural activity. And this wave of neural activity spreads throughout the nervous tissue. And because of the corpus callosum, can spread to the other side, affecting the other side as well. People who have very bad epilepsy can get these huge, massive, what they call grand mal seizures, where most of their cerebral cortex lights up with neural activity, and they basically go into convulsions, collapse to the ground, right? This is where you got to put the wooden spoon in their mouth so they don't bite their tongue off, right? Clearly, these people can't drive. It's very hard for them to live normal lives when you have this potential of at any time falling to the floor and having these massive, elaborate convulsions. The good news is, if there is good news about this, is that typically when this epilepsy occurs, there is typically one region of the brain where that occurs. Now, as Mitch pointed out, in some of those instances, that area of the brain can be removed. However, do you think there are areas of the brain? I know everybody talks about you only use 10% of your brain. Is that accurate? Are you only using 10% of your brain?
No.
No, absolutely not. Your cerebral cortex is valuable real estate. If there was a part of it that wasn't being used, parts that were being used would grow into that area. So there aren't any areas that you don't really need. So it may not be an area that can be easily cut out. However, what they can do is they can take a nice big long scalpel and they can insert it into your longitudinal fissure and they can cut the corpus callosum. They call this a split brain procedure. By cutting the corpus callosum, you stop most of the signal from getting to the other hemisphere. So these individuals still produce those neural storms, and those neural storms still spread across the one hemisphere, but they don't travel to the other ones, to the other hemisphere. And that dramatically decreases the severity of these epileptic fits and can dramatically reduce the severity of the seizures. Really good, really important, really useful. But if you get this split brain procedure, you're pretty much screwed for life, right? You can't function normally if the two hemispheres of your brain can't talk to each other, right?
Yeah.
It turns out you can. Individuals who have split brain procedures done are actually really hard to tell apart from people that aren't split brain. And the reason for that is, for the most part, the real world is static. So when it comes to clapping your hands, you kind of have an idea where your left hand and your right hand is. You may not be able to line them up perfectly, but you're going to be able to get close. If you look at an object, like if you look at a block or a book, and you see the left side of it and you see the right side of it, right? Is the left side dramatically different from the right side? No. When someone speaks to you, is the sound coming into your right ear that different from the sound coming into your left ear? No. For the most world, the world is pretty constant. In fact, for these people with split brains to actually see the effect, they actually have to do some pretty elaborate experiments. They have this viewer that they'll have the person look into. And what they do with the viewer is they have one side of the face of a male and the other side the face of a female. And they'll ask them to do two things: to write down what they see and to say what they see. And what will happen is they will say male and they'll write down female, and they won't realize the discrepancy. So yes, there is a deficiency to their function, but it's not nearly as debilitating as you might think it would be. And like I said, it dramatically improves their epilepsy. So it is definitely, again, it's a dramatic surgery, but it is one that can be very successful. And again, it's nice because corpus callosum up here, fornix right here. All right, questions on that? All right, so what's in between the corpus callosum and the fornix? Well, it turns out it's a structure called the septum pellucidum. We've used the term septum in this class before. Where did we use the term septum in this class before?
On our nose.
In the nose. It's a dividing structure. And that's what this septum is as well. Notice here, we have this funny looking transverse section where they've done it at a couple different levels. As we hinted at before, not only is there fluid, cerebrospinal fluid, surrounding the brain and protecting it, but remember we mentioned how there are spaces inside of the brain that are filled with fluid as well. This here shows the two lateral ventricles, these two spaces that are to the sides of the brain. Notice this also shows a little bit of our corpus callosum. And notice underneath the corpus callosum is this thin membrane. And this thin membrane, let's actually make that a bold line. This thin membrane, that's not thick enough. This thin membrane separates the two lateral ventricles from each other. So we have these two fluid-filled spaces with a little membrane that divides them and keeps them separate. And that dividing structure is right underneath the corpus callosum. Notice you may not have been aware of it, but if we again go back to, there it is, this picture here, notice we saw it here as well. Here is our corpus callosum. Here is one lateral ventricle and the other lateral ventricle. And this thin piece of membrane in between is that septum pellucidum. So think about what's happening in that sagittal section. When we look at that sagittal section where they've cut through the hemisphere, notice above is the corpus callosum, below is the fornix, and then there's that membrane in between. So notice if we poked through that membrane, we would be in the lateral ventricle. And that's what we see with this. This is the septum pellucidum. And if we were to take a probe and poke through there, we would be in the lateral ventricle. And notice we see the same thing in the illustration as well. Here is the septum pellucidum. And again, if we poked through there, we would be in the lateral ventricle. All right. Questions on that? All right, there is one more structure we are responsible for in the limbic system, in the telencephalon in general. And that is these three nuclei right here. Remember how with a muscle group is made of 2 or more muscles, and we didn't need to know the names of all the individual muscles. We just needed to know the name of the muscle group. Well, that's kind of what happens here. This is a group of 3 nuclei. They happen to be the putamen, the globus pallidus, and the caudate nucleus. But you don't need to know the names of the individuals. You just need to know the group name. And the group name is the basal ganglion. Now, again, there's a big, huge issue with that because as we know, a ganglion is a cluster of cell bodies outside the central nervous system. And yet anatomists, for some incredibly stupid reason, called this the basal ganglia, and you will find it in much of the literature. However, they're slowly getting better. And more you will see this called the basal nuclei. Because basal ganglia is out there so much, even though it's not appropriate, I will let you use either of those 2 names to talk about these collective structures. So basal ganglia, basal nuclei, you may call them both. Of course, the important question is what do they do? And the short version is they're your autopilot. All right. Do you just spontaneously get up, get in your car, and drive to work? No, of course not. But if you happen to get in your car and you start grooving to the music, or maybe you're listening to a really interesting podcast, or you're just daydreaming, and suddenly you regain consciousness and realize you're halfway to work, which is a problem because you were supposed to be going to the store for milk. What the heck happened there? Well, what happened is your basal nuclei. Your basal nuclei's job is to maintain a learned behavior. It doesn't start or stop the behavior, but it keeps it going. That 10-month-old, when they're learning to walk, you can almost see the smoke coming out of the top of their head as they are using every single neuron in their body to think about where they're putting their arms and where they're putting their hands and where they're putting their feet and how their legs are moving as they're walking, as they're learning to walk. Does it require that type of mental exertion on your part to keep walking for you now?
No.
No, you're texting, having a conversation, eating a sandwich, doing your makeup, all while you're walking down the street.
That's talent.
How many times, again, this happens to me all the time. How many times do you get in the shower and you're sitting in there humming along to the music, you know, thinking the deep thoughts that you have in the shower, and suddenly you can't remember whether you washed your hair or not because your autopilot kicked in, and you don't know whether or not you went through washing your body in the normal fashion that you do, but you can't remember whether or not you washed your hair or not. All of these are examples of that basal nuclei doing its job, maintaining that learned behavior. Again, you don't spontaneously start the behavior, you don't spontaneously stop the behavior, but once you start walking, it keeps you walking without having to think about it, which allows your mind to wander and do all those other things.
I have a question.
Yes.
Is that the same thing like when you're driving and you're spacing out, but you couldn't remember if the light was green or not? Because you like come back and you're like,
Yes, unfortunately, yes, exactly. So again, driving is another great example. All right, I've got a 17-year-old now who learned to drive during the pandemic. And again, when she's first in the car, right, trying to learn, it takes 100% of your focus. You're thinking about every little thing you do, but once it becomes a learned behavior, then once again, you're driving and texting and eating a sandwich and doing your makeup and doing all those things at the same time instead of thinking about the driving, because it's a learned behavior and your autopilot is keeping it in track. The problem with that is, yes, you may accidentally run that red light, or like I said, you may end up halfway to work when you were supposed to be going to the store. So that's the downside of that autopilot, is that, right, when it can not kick in at not necessarily the appropriate time. So paying attention while driving is important. So don't run red lights. All right. Questions on that? All right, excellent. The good news is with that, we have now identified and distinguished everything we needed to know in our cerebrum. And again, remember, all of the structures of the cerebrum make up all of our telencephalon. So what we need to do next is go to our other cephalons, starting first with our diencephalon. Remember, our diencephalon becomes a series of structures that in the mature brain are actually still known as the diencephalon. So that makes it easy to remember which cephalon these structures are in, because the diencephalon is the diencephalon. But we'll do this after the break. Let's go ahead and take another break. This is a good natural stopping point. It's a teeny bit early, but this is a good natural stopping point so we can come fresh. It is 11:08. So that means coming back in 15 minutes, which will be 11:23. So at 11:23, we will restart. And that should give us enough time to, again, if you look at the list, we have far fewer structures in the rest of the cephalons we need to identify. So we should be able to finish off the rest of our brainstem in today's lecture. So that should be great. All right, so excellent. We'll come back then, and I will start the recording at that point. All right, any questions before we take our next break? All right, excellent. See you in 15 minutes. All righty. So, as I mentioned, we are on to our second cephalon, the diencephalon. And I know from this illustration from your textbook, it looks kind of blobby. But one of the easiest ways to see it is to, well, actually look at it from this picture here in a sagittal section. This, it always kind of reminds me of a duck face, right? There's the bill, and here's the eye with the pupil. This region right in here, let's include a couple more structures. That is the diencephalon. And notice the most obvious structure in the diencephalon is the big, huge, bulging eye. And that big, huge, bulging eye is a structure known as the thalamus. If we go back, we actually see what the thalamus is. The thalamus is another muscle group. Like the basal nuclei, the thalamus is a collection of nuclei. And its function is basically to be a relay station for sensory information. Almost all sensory information goes to the thalamus first. Before the cortex. What's the one exception? What do you think the one sense that doesn't go to the thalamus first before it goes to the rest of the cerebrum might be?
Smell?
Exactly. Our sense of smell, because we know that goes straight to the limbic system. So actually, let's be more specific. Instead of cortex, let's say the word cerebrum. So yeah, everything except smell goes here first. And notice if you look, there are all these different nuclei with different types of sensory information. And in fact, as I mentioned, I'm a visual neuroanatomist, and a lot of my research was actually looking at this tiny little nucleus right here, known as the lateral geniculate nucleus, which is what receives its visual information. So all of our information from our eyes goes to this lateral geniculate nucleus first, before it goes to the occipital lobe to be processed. You don't have to know that. I just, as one of the requirements of my grant, I'm required to say lateral geniculate nucleus 3 times in my lectures, and now I've done it twice. So excellent. So we have that thalamus, which is that relay center. Now, notice one other thing, and I think I've got the pretty picture for it going this way. Excellent. Here in blue, we have the thalamus from one side and the thalamus from the other side. And notice the 2 thalami do not touch. There is a space in between. This space in between is our 3rd ventricle, another one of those fluid-filled spaces. But if you look very closely inside of this 3rd ventricle, you can see there is one little stem-like structure that connects the 2 thalami together. Notice I've got a thalamus on one side, I've got a thalamus on the other. They don't touch. So the space in between is the 3rd ventricle. But notice there's a structure that actually does connect them together. So that notice when we cut through the thalamus and look at a sagittal section, we can see that one little nub of that stem sticking out. It's white, which reminds us that it is white matter. And what do you think the name of this little bundle of axons that connects the 2 thalami together might be? Well, what's the second thing on your list?
Intermediate mass.
Yeah, see how easy that was? It's the intermediate mass. So there you go. On the exam then, if I ask you to identify this structure, what are you going to say?
Thalamus.
And if I point here and ask you to identify this structure, what are you going to say?
Intermediate mass.
There you go. But we have one more issue as well. Again, notice this is also a space. What space would this be? What did I just call the space between the 2 thalami?
The 3rd? Was it the 3rd ventricle?
Why is it the third? Because we have the two lateral. The two lateral are 1 and 2, and then the third is the one right here in the middle. So this space is the third ventricle. However, notice the wall of the third ventricle is made up of structures. One of those structures that makes up the wall is the thalamus. But notice there is another structure that helps to form the wall of the thalamus, the wall of the third ventricle as well. It looks just like a smooth wall. As we'll see, it's a little bit more elaborate than this. But this structure that forms the wall happens to be below the thalamus. So guess what we call this structure in the wall below the thalamus?
The hypothalamus.
Because what does hypo mean?
Below.
Below. There you go. And that is exactly how I would word that on the exam. I could have an arrow pointed into this region, and I will ask one of two questions. Ask you to identify this space, in which case you will say third ventricle, or I will ask you to identify this structure, in which case you will say the hypothalamus. Now, we can take a closer look at the hypothalamus, that smooth wall, and we see once again, like the thalamus, like the basal nuclei, it is a collection of nuclei. We don't have to worry about any of the specific names or any of the things associated with that, but there's a lot of stuff there. So not surprisingly, there's a lot going on in our hypothalamus. It plays an important role in the regulation of breathing, of temperature, of a bunch of physiological processes, stuff like that. This also is what is responsible for regulating, actually, let's go back. Controlling the endocrine system. What does the endocrine system do again?
Isn't it like signals?
Hormones.
Yeah, it is another organ system specialized for communication and responsible for producing and releasing hormones. This is actually a very surprising finding. This only occurred about 20 years ago, which is pretty remarkable if you think about it. And the reason for that is this M&M that hangs out underneath the bottom of the brain. Out of the bottom of the brain is this big M&M known as the pituitary gland. The pituitary gland is also known as the master endocrine gland. Why? Because it produces over a dozen hormones. And many of these are hormones like thyroid stimulating gland. Guess what the thyroid stimulating gland does?
Stimulates the thyroid.
Stimulates the thyroid. See how easy that is? Absolutely. Most of these hormones control the other endocrine glands. So for many, 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 many years, we believed that the pituitary gland controlled the endocrine system. And it does. However, it turns out the pituitary gland is controlled by the hypothalamus. The pituitary gland is basically like the president. It's the one that's up front, that everybody sees and everybody believes is in control. But we all know that behind the scenes, the Illuminati really is controlling the presidency. And that Illuminati is the power behind the figure. And that's basically what the hypothalamus is. The hypothalamus controls the pituitary gland, and then the pituitary gland controls everything else. All right. And of course, you know, I'm joking. There's no Illuminati. It's all aliens. All right. Excellent. But I'm sorry.
I just said, mm-hmm.
No, it's aliens. Notice one more structure. There is this stalk-like structure that connects the pituitary gland to the hypothalamus, connects the M&M to the bottom of the brain. And what might that structure be?
Infundibulum.
Right, absolutely. Which, if you didn't see it on the list, you probably could have guessed because it said it right there. I want to remind you one more thing about this pituitary gland. Oops, hold on. And I want to go back to, come on, where is it? This picture. Remember back in ancient times, we were learning about the bones. And there's this nice indentation right here. What was this nice indentation called? This U-shaped structure called?
Sella turcica.
Sella turcica, because it looked like that Turkish saddle. So it had a really high front and a high back. But remember, we also said its term was the apophyseal fossa. Apophysis is another name for pituitary gland. Fossa is an indentation. So notice it is the pituitary gland that sits in the hypophyseal fossa. There's the infundibulum connecting it to the hypothalamus. Here's our thalamus, and there's our intermediate mass.
I'm a little confused. So for some of the features, we need to know the functions, but others not.
Notice most of the things you don't need to know the functions of are white matter structures, are bundles of axons. They're just pathways for carrying information. Right? Corpus callosum, fornix, infundibulum, all of those things are just bundles of axons. They carry information from one place to another. It is the cell bodies, it's the cortex or the nuclei. That's where we process the information. That's where we make sense. Those are the things that have functions. Notice there are 3 more white matter structures, just bundles of axons that we see here. Let's start with the 2 easy ones. We know, as I mentioned, the corpus callosum is the largest commissural structure, the largest bundle of axons going from one hemisphere to another. But with a name like largest, what does that imply?
There's the smallest.
Yeah, there are others, absolutely. Yeah. And those others are located right here. One, the corpus callosum basically, or in the fornix, basically point right at. And the other is straight back from it. So I made a little bit of a mess, so let's get rid of that and that and that and that. So we can emphasize these 2 bundles of axons that go from one hemisphere to the other. Notice on our illustration here, one's numbered 73, one's numbered 78. But more importantly, one's towards the front of the brain and one's towards the back of the brain. So guess what we call the one towards the front of the brain?
Anterior.
Anterior what?
Frontal.
No, look at your list. Anterior what?
Oh.
Commissure. Yeah, remember commissure is white matter bundle goes from one side to the other. And that makes 78. Yeah, you were overthinking it. Exactly. And so
I thought you were just asking like which ways we could say it.
Oh,
like...
I see. And so then what would the bundle of axons labeled 78 be?
Posterior.
Posterior what? Posterior commissure, exactly. So notice just as simply, we have the anterior commissure and the posterior commissure. So these 3 things that we've drawn in red, the corpus callosum, the anterior commissure, and the posterior commissure are basically just bundles of axons that carry information from one side of the brain to the other. Now, there is one more white matter structure we have identified already, but may not have thought of it in these terms. And that's this structure right here in front or above the pituitary gland. And let's get a better look at this. There we go. Notice this is an anterior view of our brainstem. Notice what we see really nicely is the two thalami and therefore the third ventricle in between them. Notice this picture doesn't show the intermediate mass that would connect the two together, but that's okay. It shows the infundibulum, the stalk, but notice it doesn't show us the pituitary gland because the pituitary gland would be there and would be in the way. And we don't want that because we're trying to look behind it. Notice also while we're here, we see two structures and now we understand why they get their name. These two structures right here are the mamillary bodies. And why do you think they're called the mamillary bodies?
Because they look like boobs.
Exactly, two little globular things that look like boobs, and anatomists are mostly male. There you go, exactly. So you might have wondered about that name before, but again, you see there's two, one on each side, and that's where they get their name. But that we've already identified, now we've seen it. What we looked at before was this structure right here. Notice, as we were talking about before, you have axons coming in from your eyeballs, giving you information from the eyes, eye-specific information. But remember, we need to get to side-specific information. So to get to side-specific information, we had this special area where some of the axons crossed. And remember that area where some of the information crossed, we called the optic chiasm. Let's take another look. Here, we peel the rest of the brain away, and we can see how some of the axons coming from one side cross to carry the information to the other side, and some of the axons don't cross. Same thing for the information from the left side of the world. Some crosses, some doesn't cross. And that way we go from eye-specific information to side-specific information. So we have three different structures here. The structures that have eye-specific information, the structures that have side-specific information, and the structure where we get the partial crossing. And all three of these things get names. Eye-specific information is carried by the optic nerves, which of course we have two from. Why do we have two optic nerves?
Two eyeballs.
Two eyeballs. Our side-specific information are the optic tracts. And we have two of those. Why might we have two optic tracts?
Two eyeballs.
True, but two sides of the brain. Remember, we want to go to the contralateral sides, two sides of the brain, and one structure where it crosses. And remember, we mentioned it earlier, it's the optic chiasm. We only have one of those because that's where we get that partial crossing. So notice, if you take a sagittal section of the brain, you cut right through that optic chiasm. And that's what we see when we look here. This structure right here is the optic chiasm, right? Mammillary body behind the pituitary gland, behind the infundibulum, and in front of it is the optic chiasm. So that's that midline structure where we get that partial crossing. So again, if we think in terms of, as we look at this, let's clear the drawings. Think of terms of, you were talking about before, white matter structures that we are responsible for identifying. So far, we've got this one. What's this one? Identify that white matter structure.
The corpus.
Corpus.
Corpus callosum.
Callosum, excellent. What was this one?
The fornix.
Excellent. What was this one?
The anterior commissure.
There you go. What was this one?
The posterior commissure.
What was this one?
Wait, where?
This one.
The intermediate.
Intermediate mass. And lastly, what's that one?
Optic.
Chiasm. I
Chiasm.
don't care how you pronounce it as long as you spell it correctly. Excellent.
Does it have an A at the end?
No. No.
Because on our list of features, it has an A at the end.
No, really?
Yeah.
No, it should not.
Okay.
Yep. Sorry.
Okay.
So 1, 2, 3, 4, 5, 6. 6 white matter structures. You were talking about functions before. None of these things we really have to worry about their functions. Heck, we could even add another one. Infundibulum. All right. All of those are just white matter structures that you just need to be able to identify. We don't have to worry about the functions of any of these things. Maybe the possible exception would be the optic chiasm, where we know we're getting a partial crossing of the visual information. But if I were to ask you that, it would probably be more likely in a picture like this, or even a picture like this, right, where it emphasizes what that crossing is. And notice all of that visual information, where does it go first? Before it goes to the cerebral cortex, to the thalamus, specifically the lateral geniculate nucleus, which is now the 3rd time, and I'm clear. Excellent. And then it goes to the occipital lobe to be processed. All righty. We are almost done with the diencephalon, just that quickly. If I believe there's only one thing left we haven't identified, isn't there? What is it?
The pineal body?
Pineal body or pineal gland. This right back here is your pineal gland. I don't like that. Purple doesn't show up very good. Let's try dark red. That shows up a little better. This is the pineal gland or the pineal body. Or if you don't like that, you can also refer to it as your first chakra. You can also refer to it as your 3rd eye. Because what is it that your pineal gland does?
Your intuition?
Not a bad guess. I could see where you would get that from the 3rd eye or the first chakra. But ignore those for now. Anyone know what the pineal gland does, what it produces? Come on, I know you know, even if you don't know, you know.
Isn't it some sort of, I want to say it's a hormone.
It does produce a hormone.
Ah,
There you go. Mitch has got it. Excellent. Melatonin, which is the stuff that makes your skin dark, right?
no.
Oh,
No,
wait, what makes your skin dark?
Melanin.
Melanin. Okay. Then what the heck's melatonin?
Makes you tired.
Exactly. Melatonin plays a huge role in helping to regulate your sleep-wake cycle. We actually have a sleep-wake cycle. I know that may be hard to believe, because how many of you just jumped out of bed on your own accord this morning? Anyone?
No.
You're a sick person. No, most of us got up this morning because our alarm blared in our ear, forcing us to get up because we didn't want to get up, but we had this class at 8 o'clock in the morning. So our alarm went off at 7:55 so we could be here in time. You could take the massive commute from your bed to your chair and be able to take this. Absolutely. When we eat, when we sleep, when we wake up in the morning, all of those things are much more controlled by our social interactions and our social responsibilities. However, birds don't have social responsibilities, right? They don't have social cues. They don't have alarm clocks. And yet they're able to get up every morning at the right time and do the things they need to do at the right time and go to sleep at the right time and even migrate at the right time because they have a circadian rhythm. Way back in ancient times, and by ancient times, I mean the 70s, there was this mystical, magical place called college. Now, college is not what you and I are experiencing right now, where all this real life is going on and we're trying to fit these classes in around it. Back in the ancient times of the 70s, young men and women, after they were done with high school, they would go to a 4-year university, right? And at that 4-year university, as freshmen, they were required to take a psychology class. And that psychology class was basically an excuse for the instructors there to torture these kids. I'm sure you've heard many famous examples. Probably one of the greatest examples was the prisoner's dilemma that happened at Stanford, right? A great example of one of these social experiments they underwent. Well, one example of these is they were curious. What if you removed all of the social cues? Do humans actually have their own sleep-wake cycle? So what they did is they took a bunch of freshmen and they locked them each into their own individual rooms in a basement. These rooms had no clocks, no windows, no TVs, no way of keeping track of time. They had a fridge, they had their textbooks, they had a light switch that they could control, and they lived in there. And after about 3 or 4 days, what they found is that an individual would get on a fairly consistent cycle where they would typically wake up at a specific time, eat meals at a specific time, and go to bed at a specific time without any external cues affecting them. And what they eventually found out is it was this hormone melatonin that plays a huge role in this. During the course of our day, your pineal gland produces melatonin, and as melatonin levels get high enough, you get sleepy and want to go to bed. As you sleep, that melatonin is broken down, and you wake up bright-eyed and bushy-tailed in the morning. Now, hopefully, none of you have been locked in a basement for over a week in time, but many of you may have, again, big weekend coming up. Thanksgiving's right around the corner, so maybe you're flying to France. And when you fly to France for Thanksgiving, when you get off the plane, it's, evening, you're ready to have dinner and party. And what's the problem with that?
The time change.
Yeah, everybody in France is getting up, having breakfast, and getting ready to go to work. What do we call that disparity between what you think the time is and what they think the time is?
Jet lag?
Jet lag, absolutely. So that jet lag is that disparity in melatonin, and that's what our pineal gland is good for. Now, notice I also called our pineal gland your third eye. The reason for that is there are direct connections from your eye, from your retina of your eye to your pineal gland. So light plays a huge role in affecting the production of melatonin. One of the best ways to combat jet lag, the first day when you get to France, when you get there, wake up the first morning at sunrise, sit out on the front porch and take in about an hour of sunlight. And so just watch the sunrise for an hour, and that will help to reset your melatonin production and help you to be able to get into a normal cycle with minimal disruptions, right? Why don't you just pop that melatonin pill when you get there to go to sleep?
You can.
That's one of the worst, horrible things you should do. If you've got melatonin pills, throw them out. As I'm sure you know, and we'll talk about this more when we get to the endocrine system in 431, do you just let hormone levels become whatever you want in your body?
No.
No. We keep very, very tight control of our hormone levels because hormones play such an important role in our function of our body. So if you take a pill that massively increases your melatonin levels, your pineal gland freaks out. Oh my God, we have way too much melatonin. I need to stop producing it. And so it stops melatonin production. So that's great. That big, huge dose of melatonin helps you sleep tonight. But then all day tomorrow, you're not producing a lot of melatonin. So what happens when bedtime comes?
You're not tired again.
You're not tired. So what do you have to do? Pop another
Take
pill.
another one.
And you suppress your melatonin production even more. And then you have to pop another pill and you suppress your melatonin even more, right? Those melatonin pills are very, very troublesome for your homeostatic balance. So definitely something that you should kick if you are doing them.
I took one last night. I full attack. I never take it either. I just could not sleep. So
There you go. Well, okay. So be careful about it. Do like everybody else does. Just drink alcohol. Wait, no, I'm just kidding. Yeah, obviously, are there situations where it can be helpful? Yes, but like any hormone that you're taking, hormone supplement that you're taking, you have to think about the long-term implications that it has on your body. The other flip side of this as well. Well, and again, like I said, the problem is that we rely too much on our social pressures as to when we wake up, when we sleep, when we do things like that. So since we're not able to set our own rhythms very often, they often get disrupted. And so especially if you're working in med school where you're, you may be working a 12 or 16-hour shift and sleeping at weird times. Yeah, there can be people and there can be stressors that cause that, but we have to worry about the implications for doing things like that. So it is something you do need to be careful and conscious of. The other place where you see this, the other big change between the 70s and now is nightlights, right? Back in ancient times, when I was scared of the dark, my mom had this little thing that plugged into the outlet and it was a little light bulb that would turn on, and she'd turn it on at night and it would basically stay on all night long, right? However, nowadays, most night lights you get for young kids are on a timer. They're on for about 20 minutes and they turn out. Now, sure, some of that could be to save electricity, but more important than that, when you are sleeping in a lot of ambient light, it affects your ability to get deep, restful sleep. Light through the pineal gland affects our ability to produce deep, meaningful sleep. And so if there's a lot of ambient light, you're not able to break down as much of the melatonin. And as a result, that light stimulates the production of it and you wake up more tired. So they were finding that kids weren't necessarily getting enough deep, meaningful sleep. So now the light stays on long enough for them to fall asleep, but then turns off so that the room is more dark and they get more deep, meaningful sleep as a result of it.
in places that like are always, I guess, gloomy or overcast or don't really have much light, are the people more tired there?
Not only are people more tired there, but
depressed.
Minnesota, for instance, yeah, exactly. Minnesota, they talk about, that winter craziness that can come on people when they're not getting a lot of sunlight, when they're not getting a lot of that. Yeah, so being there in the dark for a longer period of time, it can have that kind of an effect. Absolutely.
So melatonin can be kind of like linked to depression, or indirectly?
That is a great question. I don't know the effect that melatonin plays on that. Because again, as we've seen, the visual system affects so many parts of the brain. I'm not sure if that's directly related to melatonin, but it's a great question. I don't have an answer for that one. It may play a role. I'm not aware of one off the top of my head, but that certainly doesn't mean that it doesn't. I'm not aware of it.
I feel like that would be like more connected to like where they call it seasonal depression.
Yeah, exactly. But
Yeah.
I don't think that's brought on by melatonin. In fact, we're going to talk about the reticular formation in a little bit, so it may play a role more in stuff like that. But it's possible melatonin could play a role. I'm just not familiar with them. But yeah, absolutely. All right, just that easily, we are done with the diencephalon, and here's an actual brain showing all of these features really nicely as well. Oh, here's another real brain. Notice pituitary gland, optic nerves, optic chiasm, the little boobies of the mammary glands, all of those things that we have talked about. All right, where we are on now, we did that. Let's do this. Is the next cephalon, the mesencephalon. The mesencephalon, as I mentioned, is also sometimes referred to as the midbrain because it's the middle part here of the brainstem. It is the smallest of the regions here in the brainstem. And we see it in a sagittal section here. We see it here, emphasized in green in this picture here. Notice the most prominent component, and let's go ahead and use green since they did it on the last one, is this big white matter structure here in the front. Notice on the anterior side, we have these two big white matter knuckles that are primary axons going to and from the cerebrum. This is the major pathway that most of that information from our spinal cord is taking to get to the thalamus and from the thalamus to the brain. And these two big knuckle-like structures in the front are known as the cerebral peduncles. We see it nicely from this anterior view. We see it nicely from the sagittal view, but actually the easiest way to see it is in a cross section. Notice in this cross section, we see this big, huge bundle of white matter. And again, white matter just means axons to us. On the anterior side. And again, their job is basically just carrying information to and from the cerebrum. However, it does provide a useful landmark that allows us to see some of the other features we're responsible for. In fact, if you look right behind the cerebral peduncles, is this very dark stained region. So dark stained, it looks a little bit like a black substance. Of course, they couldn't call it black substance, right? Because these anatomists are so picky. So instead, they gave it the fancy name for black substance, substantia nigra. Substantia nigra is dark stained because all that dark stain is actually, this is the primary place where we make dopamine. Remember, we talked about how dopamine plays an important role in our mood and our social anxieties and in Parkinson's. Parkinson's patients actually, when you cross-sect their midbrain, their mesencephalon, their substantia nigra is actually completely clear. It's actually the primary way they diagnose Parkinson's disease. Now, clearly, can you do that in a living person?
Hopefully not.
Yeah, hopefully not. Of course not. Of course not. Absolutely. So notice confirmation of Parkinson's is actually not confirmed until actually after death. All right. Again, we assume someone has Parkinson's based on the symptoms and everything, but the only 100% certain way to know for certain is actually to look in the substantia nigra. And so again, typically it's not confirmed until after death. Now, notice here when we look at the actual tissue, it's a little tricky to see what else is going on down here in this little Mickey Mouse looking head. Luckily, we have some illustrations that will show this as well. Now notice they flipped it around on us, but luckily Mickey Mouse has those nice big ears that make it easy to identify. And again, they've put stripes in it to help us to recognize that these are just bundles of white matter going from one location to another. Deep to that, we have our substantia nigra, that dark substance that we've seen here. But notice there's one additional, well, actually, there's two additional structures we want to be able to recognize, but one in particular we want to see from here. In this one here, there is an area known as the reticular formation. This reticular formation helps in, and this is kind of what we were talking about a little bit before, plays a vital role in our intention, paying attention to things. And again, one of the classic examples is, as I always love to use my wife for these types of examples, she's one of those people, the dog barking outside, sirens, gunshots going on outside, all of those things, she soundly sleeps through all of them. But when that newborn baby whimpers just the tiniest bit, what happens?
She's awake.
Instantly awake. Or the floorboard creaks because I'm trying to get out of bed to go play Xbox, and she is instantly awake, right? Why is it? Is that creaking of the floor bed louder than the war going on outside? No. But does the dog barking outside have equal importance to that whimpering baby?
No.
No. So notice how alert we are isn't necessarily just related to the sound of the volume or the intensity of a sound, but also the significance, the importance of this sound. And not only is this affected by sound, but also affected by light as well. As we talked about, for some people like having the TV, like having the noise, like having the white noise to kind of drown everything out when they're going to sleep. But often when you're having difficulty going to bed at night, one of the things you may be doing is staring at your phone for hours. And is staring at your phone for hours a good way to try to get to sleep? No, because that light, specifically the blue light, we now know plays a role in stimulating this reticular formation area, maintaining your alertness. So what do? They now have a nighttime setting to your phone to try to reduce the amount of blue light. Where was I at? I was at the store, I think the grocery store or something like that. And they were selling glasses. They have glasses now that kids can wear that cut the blue light from those as they are looking at watching TV or on their devices, right? Because we now understand how that can affect this reticular formation and that alertness over stimulating it. So this is all examples of this importance of this reticular formation.
I have a question.
I have an answer.
What if you use the blue light filter, but it still gives you problems with falling asleep?
Well, the two things about that is the blue light filter it reduces the blue light. It does not eliminate it entirely. And while we're more sensitive to blue light, for some people, they're more sensitive to light in general. Like I said, some people can fall asleep with the TV on and the lights on in the room without any problems at all. And some people who are more sensitive to light might need to wear a sleep mask at night to help them to be able to cut that ambient light just to help them to be able to get that deep, restful sleep. So while this affects everybody, it doesn't affect everybody equally.
Okay.
Okay. Great question. All right. One more thing we need to recognize, again, not a big area, so not surprisingly, there's not a lot to it. There are the two big bumps in the front, the cerebral peduncles, but notice there are also two smaller bumps in the back. Or more specifically, as we look at it from the back, 4 bumps. Bump 1, bump 2, bump 3, bump 4. These 4 collectively are known as the corpora quadramina. Right? Quad, 4. And the 2 on the top are collectively known as the superior colliculi. Colliculus is the singular. And the 2 underneath are the inferior colliculi. So these 4 bumps on the back of the midbrain collectively are the corpora quadramina. And we have the 2 on top, the superior colliculi, and the 2 on the bottom, the inferior colliculi. The corpora quadramina in general is an important integrative function. One of the most important things I do in the morning is find my coffee mug and pick it up and bring it to my face. It's vital for my survival, but it's also something that we take for granted. Right now, as it's sitting over here to the side, the light from my coffee mug is hitting my retina way over here on the right side. It's going through the lens and hitting the right side. However, here's the problem. My head can move. And as my head moves, where the light from the coffee mug is, does it change where it hits my retina?
Yeah.
Yeah. And it's even worse than that, because not only can my head move, but my eyes can move as well. We think of when light hits our retina, we know where something is in space. But the problem is your eyes move and your head move. So to really know where that coffee mug is in space, not only do I have to know where I'm seeing it on my retina, but I need to know where my body's position is. Because my head can move, my body can move, my eyes can move. So I need to be able to put together those 2 pieces of information, where my body is in space and vision. And by putting those 2 things together, I can easily grab my coffee mug. And while I may not be able to move my ears the way my cat or my dog do, does tilting my head or move my body change how sound comes into my ears? Exactly. And that's what the inferior colliculus does, where my body is in space and sound or audition. See, this is usually the point where we're in the classroom, and I've already talked during the break to a kid in the back of the class. And at this moment right now in the back of the class, that kid in the back of the class takes their book and drops it really hard onto the floor, making a big loud sound. And what do we all do when they do that?
Turn around.
Yeah, you take your ears, your eyes, and your body, and you orient all of it on that stimulus. So you can take all your information and process what the heck that was to make sure it's not a bear with an axe coming into the room to try to eat us. So that ability to coordinate all of this, integrate all of this information, where my body is in space, where my eyes are, and where my ears are, all onto one location. That's what our corpora quadramina does. And the superior colliculus does for it for vision, and the inferior colliculus does it for sound. All right. Questions on that? One more thing, it's on your list, but not under this point. Notice there is a hole in the center of our mesencephalon, our midbrain. And where we see this best is right here. Remember, as we talked about, if I poke through the septum pellucidum, I would be in that lateral ventricle. But on the midline, I have my third ventricle here. And I will tell you right now, in our metencephalon, between the pons and the cerebellum is where our fourth ventricle is. So notice fluid needs to be able to get from the third ventricle to the fourth ventricle. So we need a passageway for water. All right. And if you think about it, kind of like those pesky Southern Californians are always stealing water from us. What is the name of the structure that they use to steal the water from us? Really?
I don't remember, but they're like these huge tubes that go underground, basically, right?
Yeah,
They call it
pipelines.
an aqueduct.
Oh.
Here we have an aqueduct that goes from the third ventricle to the fourth ventricle. It is called the cerebral aqueduct, or because it goes to the midbrain, it's also known as the aqueduct of the midbrain. Both of those are acceptable ways to describe it. So notice we see it here in our sagittal section, but here we see it in our cross section as well. That hole, both in the real picture and also in our illustration. So another fluid-filled passageway to the fourth ventricle, which, as I mentioned here, in between the two big enlargements of the metencephalon. And I always think of the met as the T because the T's got those two enlargements to it. And that's what my metencephalon has. It has one big enlargement to the front, that is the pons, and one big enlargement to the back, that is the cerebellum. So the T in met reminds me of those two enlargements that are the metencephalon. All right. Oh, notice here, removing the cerebellum, we see that space, that is that triangle space, fourth ventricle, and a little bit of the choroid plexus that makes the cerebral spinal fluid. We'll talk about that in a minute. But not surprisingly, these two structures communicate with each other, the pons and the cerebellum. And in fact, if we look closely, there is not one, not two, but three major pathways to get from the pons to the cerebellum. Remember the two big knuckles coming up the front, way up here, were the cerebral peduncles, those big knuckles of axons going to the cerebrum. Well, these three are going to the cerebellum, so they're the cerebellar peduncles. However, you're only responsible for one of them, the biggest, the middle. So this middle cerebral peduncle is that biggest passageway of axons going from the cerebrum to the cerebellum. Pardon me, from the pons to the cerebellum. And so that is one of the structures. And again, just a white matter structure, so you just need to identify it. When we talk about the pons is this big enlargement in the front. And like most of the things in the brainstem, it is a collection of axons going up and down and back and forth, and nuclei to control things. Probably the most important thing controlled by the pons is our major center for control of breathing. There's a lot of other stuff going on in the pons that we don't really care about right now, but producing your normal resting breath is the major function of the pons. So whereas breathing is controlled by a couple places, the pons is one of the major locations where that occurs. We've already learned a lot about the anatomy of the cerebellum. Notice there's this elaborate tree-like white matter structure. What would we call this entire white matter structure?
Cerebellum.
Well, no, but specifically just the white matter, the tree-like white matter structure.
Oh,
There we go,
arbor
the
vitae.
arbor vitae, excellent. Notice, just like in the cerebrum, the cortex is superficial. Our cerebellar cortex is on top and the white matter is deep. We also learned that right in the middle of the two layers of gray matter are some special cells. What were those special cells that we would find here in the cerebellum? I know it feels like we've already covered a ton of information already, but this was the first histology slide. Remember, between the two layers of gray matter was a special cell. What was that special cell?
Purkinje cells.
There you go. And again, we have this relationship between location structure and function. What is the function of the Purkinje cells? Or let's be more specific, structural classification and functional classification.
Multiport and interneurons.
There you go. Excellent. We had names for the grooves. What was the name for the groove?
The sulcuses.
Yep. And we had a fancy name for the ridges. What was the name for the ridges?
Folium.
Folium. Excellent. Notice all of this is new and not new information, things we've already identified. There is one new thing I want to point out. How am I going to do this? I'll just erase all of this stuff because I want to keep this for now. Keep that up there. Notice, like the cerebrum, the cerebellum also has two hemispheres. The cerebrum has two hemispheres. However, notice with the cerebrum, there is this deep invagination between the two hemispheres. And what did we call that?
Fissure.
What kind of fissure?
Longitudinal.
Excellent. We had the longitudinal fissure, which was that deep indentation between the two. But notice the cerebellum isn't like that. Instead, the cerebellum has this raised pigmented ridge down the middle. Now, again, I know when you look at this thing over to the left, it looks like a pound of ground beef. But if you look over here, you can see in the illustration, they've done a little bit of a nicer job of really kind of simplifying it. And notice it looks like a long raised segmented, well, really worm, it kind of looks like. A long raised segmented worm, which is actually where it gets its name. It gets its name from the same long worm-like noodle. And what's that long worm-like noodle?
I was going to say spaghetti, but it doesn't match with the name.
There we go. Mitch has got it. Vermicelli. Right? A vermicelli is a long, thick, worm-like noodle. And so not surprisingly, this large, well, it's not large, the hemispheres are actually larger, but this raised segmented structure on the midline of the cerebellum is known as the vermis. Because it looks like a segmented worm. So this vermis is really the only new information for the anatomy. So then the question then becomes, what is the actual function of the cerebellum? Well, it turns out that the cerebellum plays a huge role, again, in autonomic processing, and really refining motor movements. Again, we go back to walking, what we were talking about before. Now, we're not talking about the autopilot. We're talking about the ability to walk in a straight line without falling down. Right? We're talking about the ability to be able to maintain posture, standing upright without wobbling. These are things that simple, basic things we think that are incredibly complicated processes that we take for granted until we've had that 5th shot of Jägermeister. After that 5th shot of Jägermeister, is it still easy to stand upright without wobbling?
No.
Is it still easy to walk that line, that straight line, without falling over?
No.
No. There are so many minor adjustments and modifications that need to be made to maintain things like posture and things like balance and things like motor skills like moving. And our cerebellum plays a huge role in helping to maintain that. Again, one of the interesting things we can see is what happens when this area is damaged. You're in a car driving along and you're in an accident, and you don't have your headrest properly adjusted. And so as a result of it, that headrest whacks you right at the base of your skull where the cerebellum is, and your cerebellum becomes damaged or stunned, non-functional as a result of that. We call this condition ataxia. So you've had that blow to your cerebellum, and the policeman shows up at the site of the injury, and you stumble getting out of the car. You wobble while you're standing there. He gives you the sobriety test and you fail miserably. And you get taken to the station on suspicion of drunk driving. Then they give you the breathalyzer test, and what happens?
You pass it?
Yeah, you blow a 0.0. You're not drunk, you're dealing with a traumatic brain injury. Now, the good news is often when there have been numerous cases of individuals who've been arrested on suspicion of drunk driving, when really they've been dealing with, that blow to the cerebellum impairing function. And they've had this ataxia. The good news is it usually goes away. But what if it doesn't? Think about what that would be like. You spend the rest of your life stumbling around like you're drunk without the fun of being drunk. Does that sound like a good type of experience to have for the rest of your life?
No.
No. So make sure your headrest is appropriately positioned when you're driving.
So they so then they lose those motor skills, but they can think.
They
normally?
don't have, it's not that they don't have the motor skills. They can stand, but what they're losing is the ability to refine it. So what happens is, what happens is you start to lean one way, your body is able to subtly compensate for it and bring you back. What would happen is you'd lose that refined ability. So what would happen is you would overcompensate and go back the opposite direction, right? So you're more likely to be less stable on your feet, less stable standing, less stable walking, right? Because you've lost that ability to refine those motor skills. Right? It's like knitting. If you've ever learned to knit, right? My grandmother tried to teach my wife how to knit. And while my wife's doing it, she has to put all of her focus on what it is that she's doing, because it's not a learned behavior. So she's having to learn and think about every single step that she's doing. But what about grandma? Does she have to give her full consciousness to it? No, she's knitting away and having a conversation, watching TV and explaining to my wife while she's whipping it out super fast, right? Or playing the piano is another great example of this refined motor skills, where you're able to play that piano because of that ability. That's a learned behavior, right? When you're first learning, you're not like that. But after you're 10, you're just floating around at it because you have that ability to refine that motor movement because it is a learned behavior. So refined motor movements, we should add that, of learned behaviors. Doesn't help you when you're first learning them, but once it's a learned behavior, then our cerebellum helps to refine them. Again, those little kids trying to walk, another good example. All righty. And I think that's it for the metencephalon, yes?
Yep.
Yeah.
Excellent. Where are we time-wise? Oh, we're at the end of our time. All right, well, that's cool. We got 2 quick things left and then we're done. Oops. Our last region, the medulla oblongata. Remember, the same way the cerebrum is the telencephalon, the medulla oblongata is our myencephalon. It's the base of the brainstem. The medulla oblongata is where all of our basic life functions are controlled. Heart rate, temperature, right? Again, it plays a role in respiration. All of these baseline functions that help to keep us alive are found down in here. Notice, as you can see right off the bat, there are a ton of cranial nerves that come off of this structure. But if we look at the picture, there's also a bunch of nuclei inside of it as well. However, as we look at it from the external view, we see that there are basically 2 main enlargements. One main enlargement is right along the midline. These 2 are known as the pyramids. These are bundles of axons. Well, we've talked about a lot of bundles of axons. So what's so special about this one? This is actually where most of the sensory crossing, well, not just sensory, most of the crossing occurs. When we touch something and it has to go to the other side of the brain, when I want to move my right hand and I start over here on the left, right, both that motor out and the sensory in, that crossing occurs here in these 2 enlargements known as the pyramids. Notice there are 2 other big enlargements as well. They are more lateral, and they're known as the olives. And these provide us with our sense of proprioception. What the heck does that mean? Well, proprioception is actually what we already talked about a little bit earlier with the corpora quadramina. This is where we process where our body is in space. Again, unless I've had too much Goldschlager this morning, with my eyes closed, I can touch my nose. Why am I able to do that? Because I know where my body is in space. Again, one of the ways it's easy to see the function of this is to understand how it works. You know where your body is in space because you have an understanding of the size of your body and where your limbs are located. And then you're a teenage boy who enters puberty. and you grow 6 inches in 3 months. All right. And then that boy's walking down the hallway where that vase that we got from grandma, who had come from, her grandma and her grandma, passed down for 10 generations. And that vase is sitting there and his arm just swings and knocks it off the table. Does he do that because that teenage boy's an asshole? Well, it could be, or it could just be because he doesn't appreciate how much longer his arms are now. And so he's more clumsy. Heck, when you're a teenage boy, you can be just walking down the hallway and fall flat on your face because you've been drinking, possibly, or again, just because you don't understand and appreciate that changes your body have undergone. It even happens in your vocal cords. Knowing how your body is located in space allows you to put tension in your vocal cords to produce speech. Now, while I'm speaking to you right now, do I know exactly which muscles I'm contracting to produce the sounds to produce that speech?
No.
No,
I'll let you do.
No, even I don't. I don't care about that. I'm a brain guy. What do I care about muscles? But I am voluntarily controlling them. However, that boy reaches puberty and his vocal cord thickens, and now it vibrates at a different rate. So when he makes those normal contractions of the muscle to produce a normal speech, does the speech come out normal? No. Their voice cracks, right? All of those are examples of ways that that rapid growth, those rapid changes, can affect our understanding of where the body is in space. And that's what the olive does. So the olive is what allows us to know where our body is in space. And that is the last structure you're responsible for. Oh, here, I got a list here for the medulla. Cardiovascular center, so heart rate, respiration rate, vomiting, coughing, swallow, hiccup, sneeze, all those basic life functions.
Are we allowed to just put basic life functions?
You should at least put 1 or 2 examples.
Okay.
All right, I apologize for going 5 minutes over, but I thought it was important to go ahead and finish this. It's kind of gotten us caught up, so we're at a good point. Any questions on this before we call it a day? All right, as always, I have office hours after, so if you have questions, please feel free. Otherwise, again, I apologize for running a little late, and I will see you guys on Wednesday.
Thank you.
Yep,
Thank you.